4 तो बज नहीं रहे शाम की, नींद आ रही होगी, राइट? डन?
A very fine morning, everyone. My name is Dikanshi Belwar, and I'm joined by my co-host, Ms. Manit Kaur, and it is our great privilege to serve as the hosts for this session today. A very warm welcome to day 2 of the technical sessions of Forensic Science track at the International Conference on Frontier Areas of Science and Technology for Sustainable Development 2026, hosted by the Department of Forensic Science under the MIT School of Applied Sciences. I hope you all had an enriching and insightful experience yesterday. Today, we continue our journey of knowledge, innovation, and scientific exploration in the forensic sciences, a field where science meets investigation and plays a crucial role in strengthening justice delivery systems. We look forward to another engaging and intellectually stimulating session ahead. It is my distinct pleasure to introduce Professor Dr. Bhupesh Kumar Sharma sir, a renowned forensic expert, academic leader, and seasoned investigator. Currently serving as the professor and dean at STT University, Gurugram, he brings over 20 years of rich experience in teaching, research, and forensic practice. His multidisciplinary expertise spans forensic science, chemistry, cybersecurity, and forensic accounting. Dr. Sharma has worked on more than 1,000 forensic cases, including several high profile investigations, and has contributed extensively to research with numerous publications and patents. Known for his strong academic leadership and public engagement, he continues to bridge the gap between scientific investigation and societal awareness, making forensic science more accessible and impactful. I kindly request Professor Dr. Bhupesh Kumar Sarma sir, to please come forward and share his insights. Please.
So how are you feeling today? एक बार मैं करा रहा हूँ सर Anybody from master? So my name is Dr. Ukesh and I am professor of pharmacy. And for the past 21 years or so, I have been the forensic expert also. I have solved more than 3,000 cases of forensic science, which includes rape, murder cases, theft, burglary, from fraud and forgery. May it be the forgery case from bank, may it be the forgery case of a property, these are most of the forms. And I believe you all are going to be forensic expert in future. Are you excited to be a forensic expert? So I have been associated currently with SGT University as a professor and dean of School of Basic and Applied Science, as well as for 15 years I have served MIT University Noida, as well as MIT University Dubai, and I'm a freelance forensic expert as well. So today we are going to discuss about the court ethics. Many of you, after doing your bachelor's or master's, when you will be entering into the field of forensic science as a forensic expert, you need to know the ethics, how to behave in court, what to do in court, and what not to do in courts. I was not taught all these things at my post-graduation in forensic science, and I find it very hard. I found it very difficult when I face these things in. Okay. Okay. Are you comfortable in English or Hindi or mix? Both? Anyway, okay. Anybody from South India who is unable to understand Hindi, then okay. Anyway. So when I entered into the court first time in 2006, I was literally crying because nobody taught me how to act in the courtroom. I was thinking myself as a king when I was entering into the court, that आज मेरा कोर्ट का पहला दिन है, मैं तो राजा बन गया. And when I entered, the lawyer asked me, the opposite counsel asked me so many questions, and I was literally crying because I do not know how to answer these questions, how to behave in the court. And it was a breakthrough in my life. It was a changing moment in my life that I have learned all the court ethics. I went to the different lawyers. I went to different judges to whom I was knowing, and I was only 25 or 26 year old at that time. And then I kept on learning. And then so many cases came into my life, and then I learned from each and every cases. But this is the opportunity for all of you to learn from my expertise, to learn from what I'm going to teach to you. And I'm very thankful to the organizer of this conference to invite me to at least teach you something, to give you the glimpse of something which you are going to use in your life. So first thing, as a forensic expert, all of you know that forensic expert is an expert under section 39 of Bharatiya Sakshi Adhiniyam. Do you know? Earlier it was section 45 of Indian Evidence Act. Sometimes there is a question in the courtroom that how you can be an expert. आप सबके दिमाग में होगा, आपको तो experience नहीं है when you will be entering into the courtroom and you will be asked the question how you are an expert. So what you will be answering? The simple answer you have to give without any hesitation that I am an expert under section 39 of Bharatiya Sakshi Adhiniyam, and nobody is going to question you again. प्लीज रिमेंबर दिस. आप सबके दिमाग में होता है ना, टीचर्स पढ़ाते हैं, मैडम हमें तो कोई एक्सपीरियंस ही नहीं है. सर आपको तो इतना एक्सपीरियंस है, आप तो एक्सपर्ट बन गए, हम कैसे बनेंगे? Every time you are entering into the court, your degree is your biggest certificate to prove yourself as an expert in the courtroom. So under section 39, we are the experts. Okay. And the definition of experts, you already know that the person who is having a special knowledge, specialized knowledge and qualification in that particular area, whether you are acting as a toxicologist, whether you are acting as a handwriting expert, caution document expert, fingerprint expert, if you have the specialized knowledge with a degree, it is written in the book B.R. Sharma nowadays that a person who is having degree and especially masters in forensic science can act as an expert, forensic expert. So if you will read that page number 242 somewhere in the B.R. Sharma old edition, so then you will know. Now, what you need to do, what our work is as a forensic expert. तो वो किधर गए IT वाले? मैं मूव कर सकता हूँ क्या? Okay, anyways, I will be louder enough. So, as an expert, our work is to assist the court. Please remember this. Our work is to assist the court. We are the guide for the court. We are not the judge, or we are not the one who is going to convince the court by our findings. We are just the provider of the assistance. Okay? The second thing, I will come to the opinion apart. The second thing, we should be providing our unbiased opinions. Many time it happens to me as well. Clients comes to me, sir इसको please forgery बता दीजिए, ये हमारे लिए अच्छा हो जाएगा. Insurance people comes to me, sir अगर इसको accident case नहीं बता के ये कह देंगे जानबूझ के किया गया, so we need not to pay the insurance claim. It happens. So we should not be biased. We should be unbiased. अगर एक भी बार कोर्ट में ये साबित हो गया ना, आपने क्लाइंट के साथ मिलके, अभी one of my student is working as a freelance forensic expert. She went to jail in one of the fingerprint case. क्लाइंट ने बोला कि उसको ये फिंगरप्रिंट सेम दिखाना है, उसने सेम फिंगरप्रिंट दिखा दिया, एंड देन देयर वाज अ चार्ज अगेंस्ट हर इन द कोर्ट रूम. देन शी वेंट टू हाई कोर्ट टू गेट द बेल. तो ये कभी नहीं करना है. डू नॉट प्ले विद योर एथिक्स. So that's why the name of the session is court ethics. Then we are scientific witness. We are witness. हम गवाह हैं. We are expert witness. ठीक है? So the way the other people are going in the witness box, we have also to go into the witness box. कितने लोगों ने कोर्ट देखी है एक्चुअल में? कोर्ट देखी है? इंडियन कोर्ट्स देखी हैं? तो कैसी होती है? Is it the same way you see in the movies? The movies one are the high courts and the supreme courts. ऐसी भी कोर्ट होती है, but they are actually the glimpse of the high court or the supreme court. But most of the time in Indian scenario, in session courts, there will be too much crowd of so many cases, and judge will be hearing all the cases in very hurry-burry. So when forensic expert comes, the lawyer will be introducing you to the jury that this is the forensic expert in so and so case, and then the chief examination, cross examination will start. There will be several questions which will take you deep into the to initiate your knowledge as well as your scientific intervention, that how much you know about that science. अभी lawyers बहुत smart हो चुके हैं. पहले नहीं था. Now AI is there, artificial intelligence is there. So what they are going to do, they are going to upload your forensic report into the AI, into ChatGPT, and they will ask, please prepare the cross questions. And I have faced this scenario last year when the lawyer was so much. I was thinking that lawyer doesn't know anything about this particular ballistics, but lawyer has asked me so many questions. Then I thought that he might have used any AI tool or anything, because these questions otherwise cannot be asked by a lawyer. So, but we are a scientific witness, so we need to be abide to this scenario that we are only the scientific witness, and our opinion must be evidence based. Our opinion should be reproducible. What do you mean by reproducible? Any or every time the test is run, the result are going to be the same. If you have given a DNA report, okay, anytime the sample can be run to analyze the DNA and to compare the DNA with other person's DNA, and the result should be same, whether match or mismatch. As well as in handwriting or fingerprint, the reproducibility there. It should not be that somebody else is reproducing, but they found the different results. It should not like be that. So be aware of this situation. And then our report should be defensible. We should be defensible in the courtroom. We should defend our say report, whatever we have written. We should not say that this is our guesswork or this is our assumption. This is our merely an assumption, and I don't know how these results came, but I have run the test and these are the results. I should be defendable. Okay. So we need to take care of these things. Do you have the subject of law in your graduation? You don't have. Okay. They have criminology. Okay. So, first of all, there must be integrity that you need to take care. I will explain each and everything later on. You must be impartial. Okay. You must be competent enough in your subject area. Okay. Then you should maintain confidentiality. So, what happened? I will like to share one example with you. मैं नया नया एक्सपर्ट बना था in 2007 or 8, and I was handling a ballistic case. I have given a report in ballistics that whether this bullet has been fired from this particular gun or not. There was a report from FSL also that this bullet has been fired from this particular gun. I was in the defense. I was doing this case for my client, the one who was the murderer actually. अब पूरा केस मैं आपको बताऊंगा तो अ पर्सन हैज मर्डर्ड 4 पीपल. वो बस में चढ़ा, उसने 4 लोगों को गोली मारी, एंड दिस हैज हैपेंड. सो द क्लाइंट केम टू मी, प्लीज सेव अवर चाइल्ड बिकॉज वी हैव द ओनली चाइल्ड दिस. एंड तो मैंने कहा इसने तो गोली मारी है, इसको मैं कैसे बचा सकता हूँ. देन दे टोल्ड मी दैट हर हिज सिस्टर हैज बीन रेप्ड बाय दोज 4 पीपल, और इसने चढ़ के, क्योंकि इंसाफ नहीं मिला तो इसने चढ़ के गोली मार दी. तो मेरे को लगा कि ये अच्छा काम है अगर मैं इसको बचाता हूँ, क्योंकि अगर मैं भी भाई होता उस बहन का तो मैं भी शायद यही करता ऐसे ही. सो एथिक्स बीच में आ जाते हैं. तो मैंने उस एफएसएल की रिपोर्ट में आई फाउंड आउट सो मेनी शॉर्ट कमिंग्स, एंड आई एक्सप्लेन दैट हाउ दिस बुलेट इज नॉट फायर्ड फ्रॉम दैट पर्टिकुलर गन, दो इट वाज एक्चुअली फायर्ड. And I was telling this case. I used to come from, say, by train from Agra to Delhi when I was in Amity. So I was discussing this case with my fellow mates, say with my fellow travelers. बहुत दोस्त बन गए थे ट्रेन में आते जाते, तो मैं उनके साथ डिस्कस करता था केसेस. ऐसे ही मैं डिस्कस कर रहा था, और ये कैसे हुआ, एक आदमी सामने बंदूक लेके बैठा था सीट पर, अपनी राइफल लेके वो जा रहा था कहीं, तो बंदूक देख के मैं केस डिस्कस करने लग गया. केस डिस्कस करने के बाद मुझे पता चला, वो आदमी ने मुझसे पूछा कि कौन सा केस देख रहे हो आप. मैंने उसको बताया कि ये केस देख रहा हूँ, कि मैं अपोजिट पार्टी का मैं हूँ मेंबर. उसके हाथ में एक बंदूक थी. मेरे को पहले लगा ये मजाक कर रहा है. उसने सबका नाम ले दिया कि आप ही हो वो भूपेश, जो ये जिसने हमारे खिलाफ रिपोर्ट दी है. अब मैं डर गया. उसने मेरा गरेबान पकड़ लिया कि मैं आपको बताता हूँ. एंड यू कैन इमेजिन माई सिचुएशन इन दैट कंडीशन. सो यू मस्ट बी कॉन्फिडेंशियल. सो व्हाट आई डिड, मैं अगले ही स्टेशन पर उतर गया. ठीक है. यू शुड नॉट डिस्कस एनीथिंग विद योर इवन फैमिली और फ्रेंड्स, वट एवर केस यू आर हैंडलिंग, बिकॉज यू डू नॉट नो वेयर इट कैन गो. अभी एज ए प्रोफेसर व्हाट हैपन्स टू अस मोस्ट ऑफ द टाइम, हम बच्चों के साथ केसेस शेयर कर देते हैं व्हाट्सएप ग्रुप पे. हमें करना पड़ता है बिकॉज दैट्स व्हाट दे आर पेइंग फॉर. एंड इफ दे आर लर्निंग फ्रॉम एन एक्सपर्ट, वी आर अबाइड टू गिव देम द रियल केस एग्जांपल्स एज वेल. तो केसेस शेयर कर देते हैं, अब वो आगे कहीं और शेयर कर देते हैं। तो ऐसे ही फिर से हुआ इतने सालों के बाद एक्सपीरियंस के, वो बच्चे ने कहीं आगे शेयर कर दिया और फिर से मेरे ऊपर ब्रीच ऑफ कॉन्फिडेंशियलिटी एक तरीके से आ गया। तो फिर मैंने अभी भी लोगों का नाम छुपाना चालू कर दिया रिपोर्ट में बच्चों को शेयर करने के लिए। बट यू शुड बी कॉन्फिडेंशियल अबाउट द केसेस यू आर हैंडलिंग, and you must be accountable. You must follow all the protocols and procedures. Your protocols and procedure must be not only dictated but well written in the report, that what procedure you have followed. If you have done the handwriting examination, let's say it is an examination of differentiation of ink. किसी को पता है ink differentiation कैसे कर सकते हैं? If there are two types of inks, how you can differentiate? With the help of IR. यूवी लाइट एंड आईआर। आपने कभी लैब में गए होगे, आपने वीएसटी देखा होगा, देयर इज अ डॉक्यू बॉक्स आल्सो। सो आई शुड राइट दैट आई हैव यूज्ड इंफ्रारेड रेडिएशंस, which infrared radiation, what was the wavelength and how I have used. I should be very much precise in my protocols because I will be accountable in the courtroom. ठीक है, आप कुछ भी लिख देंगे क्लाइंट के लिए, खुशी खुशी का माहौल हो जाएगा, ऐसा नहीं चलेगा, because you are the one who are guiding the court. Okay, so you should be accountable and you should be very objective. You should remain neutral. You are not fighting for your client, you are fighting for justice. Please remember this. जब आप एक्सपर्ट बन जाएंगे, आपके पास ऐसे बहुत सारे मौके आएंगे when the people will try to bribe you, people will try to buy you. Okay, you will have enormous, say, plenty of chances like this, but you must be objective that what you want to do with your career. So let us explain the objectivity and impartiality. Objectivity here means focus on the fact without personal influence. पर बहुत समय ऐसा होता नहीं है, we get influenced. अगर आपकी टीचर आपको एक हैंडराइटिंग का सैंपल दे रही है कि प्लीज कंपेयर दिस, और टीचर कहती है यू नीड टू गिव योर ओपिनियन एज दिस इज अ केस ऑफ फॉर्जरी, तो आप इन्फ्लुएंस हो जाते नहीं हो जाते कि ये तो फॉर्जरी का केस है. यू नेवर ट्राई टू look at different angle that this might be a case of disguise or this might be a case of genuine signature. You always try to look for the forgery. Many clients calls me, sir ये हमारे साथ फर्जीवाड़ा हुआ है, we have been cheated, please give your opinion. So I have been influenced as per the statement of the client. I must have been influenced that ये forgery है, फर्जीवाड़ा हुआ है. Why should I be? I should be very objective in my observations. Clear? Then I should be impartial. पहली बार में जो भी सच है वही बताओ क्लाइंट को. अगर आपको कुछ गलत काम करना भी, आई एम नॉट कंपेलिंग यू टू डू एनी रॉन्ग वर्क, ठीक है, पर आपके पास एज ए फॉरेंसिक एक्सपर्ट दोनों तरीके के केस आएंगे. देयर विल बी द केसेस विच आर एक्चुअली ट्रू, देयर विल बी द केसेस विच आर एक्चुअली फॉल्स, एंड यू विल बी फाइटिंग in favor of the client or against the client, against the. Hello ma'am. Good. Yeah. So objectivity and impartiality are not just the principles, they are your professional responsibility. So you should be responsible enough. If you have any question, you can raise your hand in between and ask me. Now, integrity and honesty. What do you mean by integrity? Yeah. Be loud enough, yeah. You can give this. Just be. It's okay.
Good morning, sir. Like you talked about the case of the guy you rescued, who shot 4 guys. So in that you said that you defended him when he used when he used the same gun. But how are you able to do that?
Okay, so this is a very long procedure. In which semester you are?
I am in second semester.
Second semester. Have you studied ballistics?
Not yet.
Okay, sit down. Thank you. So, I'm asking you a question. How many of you know about ballistics here? Okay, you have studied. So you need to look for the firing pin mark. You need to look for the chamber marking. You need to look for the breech face marking and everything. Okay. Everything was clearly mentioned in the report that these breech face marking, these firing pin mark belong to that particular gun. But I asked the expert in the courtroom during the cross examination which microscope you have used to analyze these example, these examination. Now the expert was not knowing. Expert simply says stereo microscope. Now which stereo microscope? What was the magnification of that particular stereo microscope? So we need to make this as a case that he is not the expert who have actually done the examination, or this examination is fishy. He has just written the report under the influence because he was not knowing the protocols, what protocols he have used. Okay, so this can only happen when you have the good knowledge of the subject. You will read the report 3 or 4 times and then you will be able to identify the shortcomings. Clear? Thank you. It was a good question. So your findings should be say truthfully reported in your report, in your forensic report. You should be presenting results accurately as they are. Do not be afraid of that if I will not write this, my client will not my client will not be giving me money. Welcome, sir. So let the facts speak. Okay, you should not be afraid of anything. Be scientific in your report, be precise in your report. मैं बार बार ये बोल रहा हूँ, you will get number of chances that person will bribe you in n number of ways. Okay, they will ask you, they will give you 2 lakh rupees, just tweak the opinion slightly, that whether it is mismatching or matching of a fingerprint. Okay, so never do this thing. Okay. Please avoid any overstatement that this is the 100% result. You will be asked, I have been asked number of times, is handwriting a 100% science? What will be your answer? I have been asked by the court, is handwriting science a perfect science? Is it? Is it like fingerprints or DNA? And why so? Changes. And also, no person can write two things exactly alike. Whenever you are going to write your name again and again, it will be having natural variations. So this subjectivity is there. So do not overstate anything as 100%. That, sir, I am 100% correct. No, there are chances I may not be correct because I have done the examination with my full zeal and dedication using all the tools, but there may be different opinions by different expert. Clear? Then you must mention the limitations of the methods, what you have used. ऐसा कोई भी method नहीं, there is no science which is called as perfect science. कोई वर्ड में ऐसा नहीं. So never use the word perfect, 100%, all these things. There are limitations to each and everything because your subjectivity, your opinion can be different from my opinion. Okay. So integrity is doing the right thing even when no one is watching. You should be well aware about your evidence, your analysis. If you think that nobody is watching, I will write anything, I will paste the picture in the report so as it seems to be a match or mismatch. It is going to be against you because in court, some cases are solved after 10 or 13 years or 14 years. I have been summoned in a case which I solved which I have given opinion in 2006, and the case came back. I went to Dubai for 9 years. I came back from Dubai. I have served in SGT University. Last year, in 2024, I got the summon for that case. So it can be any time because this is Indian scenario. A case can go for lifetime. लोग मर जाते हैं, विक्टिम मर जाता है, कल्पिट मर जाता है, पर केस चलता रहता है. इंडिया में ऐसा भी होता है. ठीक है? Please take care of these things. The next thing is competency as well as expertise. You should only give opinion in the area where you are competent enough. If I am not a cyber forensic expert, even though I have studied the cyber forensic, but I have never handled the case of cyber forensic and I am not a good cyber forensic expert, I should not deal in cyber forensic at all. अगर मैं ये सोचता हूँ, if I am thinking that I am a forensic expert, I have knowledge of each and everything, it is a wrong thing because during the cross examination or during the examination by another expert, I may fail. So be strict. Stay in line with your expertise. Somebody is toxicology expert, be toxicology expert. Somebody is caution document and fingerprint expert, be that. Somebody is forensic medicine expert, be that. Okay, do not try to involve yourself in other incompetent areas. If you have done a degree, if you have done a certificate, diploma, and you are well aware with your expertise, then do it. Otherwise, don't do it. I often get many cases in audio video examination, but I am not an expert. I'm not competent enough to give opinion. So I clearly say I'm not competent. It is not like you should take each and every case to earn money. You should take care about your competency, because this is what it will be challenged later on. Okay. So अभी आपको पढ़ाया गया होगा कि you are forensic expert, so you can give any opinion, but there are specialization in your masters, and then you can specialize yourself in different areas with different certification and diploma programs as well. But be competent enough in whatever subject you are given opinion. Competence build confidence. I am confident in my subject area. I am the expert of caution document, fingerprint, and ballistics. I am confident in that particular area, and this build say confidence in me. Every time I go to the court, I am more trust me that this is the best expert in this area to be trusted on. If I now hope for a situation, or suppose a situation is there, I am giving expert opinion in each and every area. Can it be possible? Can it be possible I can give opinion in each and every area? Okay. I was involved in Disha Salian case, Sushant Singh Rajput's manager. Okay. I have to give opinion on the post-mortem findings, the post-mortem report. I have to give the counter report. The meeting has been left. Are you getting something to know or not? We have experts sitting there. My group. आगे जोड़िए। मैं बहुत सालों से सबको जानता हूँ। अनुभव में है। Okay. We have been doing each other for last 21, 22 years. We are the expert in their field. So you are very fortunate enough to have such experts with you. Is it fine? Okay. Now confidentiality, I have this.
गुड मॉर्निंग सर। सो आई वुड लाइक टू आस्क सर, जैसे आपने बताया उस केस में आपने उनसे ये क्वेश्चंस पूछ लिए कि आपने कौन सा माइक्रोस्कोप यूज़ किया था, कौन सा मॉडल यूज़ किया था। तो व्हाट इफ लाइक उस टाइम पे उन्होंने आंसर नहीं दिया, दैट वाज द सिनेरियो राइट। सो नेक्स्ट टाइम लाइक दे कैन डिमांड अ डेट एंड देन लाइक दे कैन रिपीट द प्रोसीजर। नो यस सर। Yes.
He has been summoned for the cross examination.
Yes sir.
It is, say there is nothing wrong in saying that I do not know. उसने बोला I do not know, but it was a win situation for us. But he has to say this. If he says something wrong, he should keep in mind that I may be an expert who is knowing this, that which type of microscopes are available in forensic lab from which is it coming. Okay, so that was my thing to ask. Okay. You need to answer then and there. अगर आप क्रॉस एग्जामिनेशन के लिए तैयार नहीं है, you should immediately tell the court, I am not feeling well today, I will come another day. It is not when the cross examination has started. After 2-3 questions, you are making the charges that sir, I am not feeling well, I am not prepared. It's a not easy situation. However, the counsel can say, the court, the lawyer can say that the expert is not feeling well today. But this again goes against. The judge knows everything and what is happening. See, court works on evidence. Here. So confidentially, I have explained you a lot. Now, common ethical challenges. What are the common ethical challenges? We often get influenced by our client. एक मेरे पास कल केस आया, मैं आपको बताता हूँ। The person said, सर एक वसीयतनामा है, बिल if there is a will, I have been cheated, sir, मेरे साथ cheating हुई है और मेरी मदर के किसी ने ये फर्जी सिग्नेचर बना दी है, please help me. So what is my state of mind now? There is a will which have been forged. Okay, so what I will do as an expert, I will look those signatures as a case of forgery. So what will happen? My all intentions, all consciousness, this is known as cognitive bias. My all intention, all consciousness and subconsciousness will go to look forward the signs of cheating, to signs of forgery. This is a mistake. And when I realized that yes, it is a case of forgery, there has been a traced forgery which has been done. And then I told the client, yes, it is a case of forgery, but I should not be influenced by the client's version or the court's version even. कोर्ट में लॉयर इतने खतरनाक होते हैं, मैं आपको डरा नहीं रहा हूँ, बट इट्स द एक्चुअल कंडीशन. आपको पसीने आ जाएंगे. But अगर आपके पास expertise है, if you are competent enough in your subject, and if you are poised and calm, आप बहुत शांत भाव से जवाब दीजिए. Take time to answer. Your everything is being recorded. आजकल तो कोर्ट में सीसीटीवी कैमरा भी होता है, रिकॉर्डर भी होता है और स्टेटमेंट भी वो स्टेनोग्राफ पास में बैठा आपका रिकॉर्डिंग कर रहा है सारा. And then you need to sign your statement whatever you have said. एक बार जो बोल दिया, then you cannot change it. So take your time. Take a minute or two to think what you need to answer. Okay, there are certain leading questions in the courtroom जिसका आंसर खाली हाँ या ना में देना होता है. एक केस बताता हूँ आपको. I have sent the report in a sealed envelope to my lawyer. I should actually send that report to the court, but I have not sent that to the court. I have sent it to lawyer. So, A4 yesterday I was in court. So the opposing counsel asked me, आपने report court को भेजी या lawyer को भेजी? I know what can be the influence of this, but I need to be truthful. अगर मैं बोलता मैंने कोर्ट को भेजी थी, they would have asked me the receipt, speed post receipt or whatever. But I was not having. तो जितना मैंने किया वो सब गलत हो जाता. So I should be very much truthful. मैंने कहा मैंने अपने लॉयर को ये रिपोर्ट बंद लिफाफे में भेजी, तो वो वहाँ पे लगी हुई है. Okay, be truthful. Don't be afraid that you will lose the case. You are not here to lose or win the case. You are here to assist the court. ठीक है ना? अगर आप जीतने के लिए और हारने के लिए लड़ने लगते ना, तो आप जल्दी जेल की सलामती की करते हो. Because then you will be influenced. Don't get influenced. Be stick to your findings, actual scientific findings. Let us come to the common ethical challenges. Pressure from the investigator. कभी-कभी पुलिस बहुत प्रेशर लगाती है. सर जल्दी कीजिए, सर जल्दी कीजिए, सर जल्दी कीजिए. जल्दी में क्या हो जाता है? इस जल्दी में क्या हो जाएगा? गड़बड़ हो जाएगी. और ये कुछ तो गड़बड़ है वाली गड़बड़, ये बहुत बड़ी गड़बड़ है. इस जल्दबाजी बहुत है. Take your time. Ask for. अगर उनको इतनी जल्दी चाहिए, you will go to other expert. We are not here. तो कुछ लोग कहते हैं मुझसे सर प्रोसेस का रिपोर्ट चाहिए. It cannot happen. It cannot happen at all. Even they will pay 10 lakh rupees, I cannot do it. So pressure to support a particular theory or expedite the results, this can be there. Then cognitive bias, I have told you there can be cognitive bias if your personal beliefs or assumptions can influence analysis. I believe that this is a case of forgery. Now this belief can actually influence the actual analysis. Okay. So stay ethical. Time constraint can be there. This happens a lot of times with the government experts as well. There are time constraints, penalty of cases are there. In that time constraint, what most of the experts do, government experts do, but madam they correct me. Either they will give no opinion report. The no opinion can be given to expedite the dependency process. The dependency channel को clear करो. Or there are some experts, some forensic experts, the body forensic experts, those who have recently joined the FSL, they may make a mistake. और ऐसी mistakes का डिफेंस एक्सपर्ट को बहुत फायदा होता है. मैं ये नहीं कह रहा हूँ कि मेरे बच्चे हमारे बीच में काम कर रहे हैं, पर ऐसी गलती का हम डिफेंस एक्सपर्ट को बहुत फायदा होता है. जो मैंने आपको भी बैलिस्टिक का केस बताया, ऐसी गलतियों को बहुत फायदा होता है. Be aware of that. Then there can be misinterpretation of the data as well. So the key principle is stay independent. Your duty is to be truth and to the court. Okay, your duty is not to be client. Your duty is not to be investigator. Your duty is to is for the truth and for the code. Stay objective, let the evidence speak, no assumption at all. एक केस में आपको बताता हूँ मेरे अपोजिट में मेरा ही स्टूडेंट था कोर्ट में पूछा इज दिस योर रियल वर्क और योर गैस वर्क उसने कह दिया दीज आर माय assumptions और कोर्ट में उसके मुंह पे कोर्ट मार मुझे बुरा लगा कि मेरे स्टूडेंट ने ये बोला ठीक है उसको समझाया एंड इट हैज बीन नाउ मोर देन 15 years he is a very big expert in our group he is earning more than 20 to 15 lakh rupees per month he is a very good expert okay stay transparent document everything disclose and communicate clearly disclose to who disclose to the authorized people do not disclose to unauthorized people never disclose anything to unauthorized people stay accountable so ethical awareness today credible justice tomorrow so this is the takeaway it's not working ये zoom का वो आ रहा है it's okay इसको बंद कर दे zoom हाँ
Ma'am, kindly share this PPT on Zoom platform also.
Being connected. There's some network.
ये
issue.
इंटरनेट निकला हुआ
Yes, sir. Wi-Fi. Srinivas ji. So slides are moving.
okay we have already discussed this please avoid any type of ambiguous language. It seems to be a loop pattern. आपको फिंगरप्रिंट बता सकते अगर मैंने ये लिखा it seems to be a loop pattern. What does that mean? It can be a loop pattern. It cannot be a loop pattern. So this is ambiguous language. It seems to be a. Either it should be because fingerprint sign is conservative sign. Either you can give opinion or you cannot give opinion. If the fingerprints are smudged, no opinion can be given. If the fingerprints are clear, clearly it should be mentioned whether it is a loop pattern, whether it is a word pattern, whether it is arch pattern, what type of pattern it is, whether it is comparable or not comparable. There should be no ambiguous language, likely to be, or it can be possible match. It is seems to be consistent with. These type of language are never required in your report because these can play a vital role in your career. Okay, to bring down your career. Overgeneralization. If I only mention it is a loop pattern. What I am saying, how many people will be having a loop pattern? Thousands and lakhs of people will be having loop pattern. This is generalization. I should mention that what are the minutiae or what are the characteristics which are actually matching in the pattern along with the loop pattern. So you must go along with the principles of ethical reporting. Your report should be complete. Okay, and your report should be clear, it should be objective, transparent, and responsible. Now, the problem with selective reporting means I have made my mind to prove this a case of forgery. Now, this is selective report. I will mention each and everything about forgery, but there are chances, as I told you, that no science is 100% perfect. Because there are chances, science can be perfect, but there are chances the way, the protocol, the thing which I have followed, the analysis, the way analysis I have done, I might have made a mistake. So the influence should not be there. Okay. Okay. Now, what you should do, what should people not do? You must stay neutral. You must be prepared. Please go prepared in the courtroom. Read your case 2 times, 3 times, 4 times while entering into the courtroom because there are questions regarding your case only. Okay. First, there will be questions regarding your qualification and expertise. Then there will be the question from the case only. And please speak clearly. And what you should not do, you should not be defensive or offensive. Okay, just answer the query that has been asked by the court. If you are not agree, to clearly say, I do not agree to this. Court will give you time to explain that why you do not agree. Okay, You should not be giving the answer as a guesswork or your assumption. Then you should not be taking sides. So I'll move. Okay, that's. Now what you should do, let's come to this. Your dress speaks everything. The way you dress up in the courtroom. ठीक है? आपने काम में जूते पहन के बैठ गए और
Sir, if possible, please speak from the podium because your sound is not coming from the cordless mic. Sir.
Okay. So you should be well dressed up in the courtroom because your dress speaks everything. Your expertise is depicted from your dress code. And then when answering questions, you should respond to the jury. Many times you look at the client, you look at the lawyer. No, it is not the way. Answer should be to the jury. The question may come from the lawyer, maybe defense lawyer, maybe prosecution lawyer. The answer should be given to jury by looking into the eyes of the jury that this is your answer. You should not look here and there. And then sit up straight. It's not working. Okay. Pause before giving your answer. You should think before you give your answer. Do not be in any hurry. Do not be in any hesitation or agitation to give any answer that you know the answer and you are going to give it correctly. Please pause. Try to avoid ums and ahs. ये ऐसा नहीं चलेगा हो सकता है सर ये नहीं चलेगा कोर्ट में okay that's why we are telling be prepared and then you should present a professional demeanor. Be professional to your expertise or to your work and then do not fidget here and there. Do not swing. ऐसे 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 करके आप स्विंग कर रहे हो कोर्ट में दिस इज नॉट द प्रोफेशनल मैनर. ये भी आपको ध्यान में रखना है. ओके. अवॉइड एनी एनिमोसिटी. एनिमोसिटी मीन्स एक ये भी है आपके पास लॉयर अगर आपको झगड़ा कर लिया लॉयर ने लड़ गया तो आप भी लड़ोगे. ओके आप भी लड़ना चालू कर दोगे. इफ यू विल बी आर्ग्यूइंग द लॉयर और एनीथिंग देन इट विल बी द कंटेंट ऑफ द कोर्ट. सो यू शुड नॉट फॉल in such type of things. You should be fair enough, okay, to while answering your question. Listen to your speech, your pace, that what pace you are speaking, as I have speaking to you very clearly and everything explaining. This should be the pace and speech in the courtroom. अगर आप बहुत जल्दी जल्दी भागोगे तो कोर्ट की समझ में नहीं आएगा, वो भी आपके अगेंस्ट जा सकता है. Then concentrate. Pause to clarify the concept. You will be given a chance to clarify your explanation. Okay, there will be leading question, yes and no, as I told you, but explanation can be given later on. Okay, do not try to explain each and everything in each and every answer. And remember everything and then respond directly to the questions. And with this, I would like to thank you. There is an email ID. You can reach out to me. So your expertise matters, your integrity build trust, your impact is real. Our impact as an expert is real. It is going to change of the life of a person. Either he might be convicted, he might be acquitted. It depends on the report and the convincing power you are having, and then your commitment upholds the justice. And with these words, thank you very much. Any questions you have? Any other question? Yes.
So we all really liked your presentation, the way you presented it, and the way you shared your overall experience throughout the presentation. So is there any way like we can connect to you? Like you mentioned the
My
Gmail
email
and all.
ID is there.
Yes, the email ID. But if there are other way like the group or Instagram or LinkedIn,
Your teacher
that
is there, Dr. Priyanka. She will get you connected with me.
okay, sir. Okay, thank you, sir.
Thank you.
Thank you, sir, for that valuable insight. Now I would like to request all of our esteemed dignitaries to please step forward and grace the stage for the felicitation ceremony.
I request Professor Goria sir to felicitate our today's guest, Dr. Anupa Rena and Professor Bhupesh Kumar Singh. I request faculty members please come on the stage. Mamta ma'am, Tapeshwar sir, Asim sir.
Moving forward, I would like to invite Dr. Bhuvnesh Yadav ma'am to please come up on the stage and introduce us to the session chair of today's technical event, that is Dr. Anupama ma'am.
Good morning, all. It is a moment of great pride and personal honor to welcome Arena ma'am. She's my mentor, she's my guide and my lifetime coach. So anything I doubt, I call her and I ask whether this is correct or not. So every time she's helping, Dr. Rena is currently working as senior scientist at a super specialty division of forensic pathology and molecular DNA laboratory at All India Institute of Medical Sciences, New Delhi. And she is working on various aspects of forensic genetics that include DNA fingerprinting, whether that is chimerism, whether that is triallelic pattern, or the gene pool sharing in siblings. She is involved in many medico-legal investigations, whether that is a Godra case, that may be the Shopian case, that may be Nithari case, or I can name many more, or mass disaster, that is Odisha train tragedy that was there. So she is involved in all these things. And she's among the first in India to handle criminal cases involving DNA fingerprinting and has contributed in thousands of cases for DNA profiling. And she's now working with some national and international projects involving DNA fingerprinting. She has taught many students, awarded many PhDs, and have many academic sessions, whether that is in India or abroad. And On personal note, I consider myself extremely fortunate to work with her and work with her under her guidance. So I request Rena ma'am to kindly come on dais and deliver her lecture. Please come on stage, ma'am.
Good morning, everyone.
Good morning, ma'am.
I'm really thankful the organizers who have invited me on this dais today to deliver my talk. I thank Dr. Bhunesh for her kind words. Let me also share of all the students, whether a PhD, MSc, or MD, she has been most honest and hardworking student of mine. And not only she calls me for any help, I even call her for my help, especially when I have to do the statistical analysis for my research papers. To be honest, I just called her maybe 2 days back. I required to do a statistical analysis in one of my cases, and she's been very helpful. And many a times, if I don't have time to find the research papers, she's the one who helps me in finding the manuscripts or the research papers for me. And she's been very helpful to me all the time, though she has left AIMS for so many years, but still I still consider her as a very good student of mine. So I start my talk. You all have heard about the DNA fingerprinting, which has DNA, but mostly you are all undergraduates. Most of you are undergraduates, and you're very well aware what you have studied in 8th, 9th, 10th, 11th, 12th, that DNA and how DNA works in our body. So I won't repeat all those things. I won't go into the basics of the DNA. That will be a repetition of your syllabus. But now this DNA has a lot of applications in our day-to-day life. Maybe clinical diagnostics, or maybe in forensics, especially. It has many other applications. As mentioned by Dr. Bhunesh, I have been working on the different research projects, and one of them has been the chimeras. And I'll be talking on the most preferable biological exhibits for the chimeric patients. We all have heard about the FSLs and CFSLs doing the DNA fingerprinting technique especially they do it for the forensic application. I find myself privileged to be working at AIMS as a hospital because
Kindly reshare the slides because it's not moving on online. You are on the third page, but I am still in the first page. So just reshare this slide, please.
So I'm presenting my talk on the most reliable biological exhibit. In forensics, the IO, he submits any kind of biological exhibit for the forensic analysis. But when I'm talking about the chimerism, we really need to know what is the best sample to be used for the chimerism. I had undertaken a research project I'd taken a research project, 2 or 3 research projects. So as Ali says, you're not a victim for sharing a story. You are a survivor setting the world on fire with your truth. As Dr. Bhupesh also says, he mentioned in his talk that you have to be very honest. You have to work for the science, not who wins or who is defeated, right? So you have to, and you never know who needs your light, your warmth, and reaching courage. Now, science, you are all the science graduates here. Science is like a magic, it's but real. And science knows no country because knowledge belongs to the humanity, and this is a torch which illuminates the world. Science is the highest personification of the nation because that nation will remain the first, which carries the furthest the works of thought and intelligence. And science, my lad, is made up of mistakes, but they are mistakes which it is useful to make because they lead little by little to the truth. Now, when we talk about the forensic science, it offers a great potential and it draws on almost every discipline. You have forensic chemistry, forensic physics, forensic mathematics, statistics, forensic biology, and many other aspects of the forensics. And it creates widespread opportunity for innovation. And biology is a part of science, is the most powerful technology ever created. And DNA is the software, proteins are the hardware, and cells are the factories. With genetic engineering, we are able to increase the complexity of our DNA and improve the human race. I don't know how many of you have heard about the dolly sheep. We tried to develop a dolly sheep, but we couldn't do it repeatedly because of the ethical reasons. We have no ethical clearance for such things because if we have a clear ethical clearance for such things, I'm Anupama standing here today. After my death, there will be somebody who wants to have another Anupama, so I'll be again here. So the same way, we had our late Prime Minister of India, late Srimati Indira Gandhi. She's no more physically with us today. But if I have an ethical clearance, I can have her with me today also. So we don't have it's not for good elements, it's for bad elements also. But so we have no ethical clearance for such things. But yes, biology or DNA, it has helped us in the genetic engineering, in the diagnostic, say, applications. Maybe we have the certain DNA or the certain genes, which our scientists, and maybe in USA particularly, they have tried to repair the cancer genes or many other genes for many other diseases. So it is where we require this technology in the clinical side also. The nitrogen in our DNA the calcium in our teeth, the iron in our blood, and the carbon in our apple pies were made in the interiors of the collapsing stars. And we are made of this star stuff. And this DNA is often referred to as the Eureka moment that transformed justice by enabling the identification of criminals and exoneration of the innocent, moving science from slow, painful slog to astonishing. I had a privilege to meet Professor Alec Jeffreys, the Nobel laureate, who established this technique long time back in probably 1994, or sorry, 84. And we had his first paper in the Nature in 1985, I believe. And I had a privilege to meet him personally in 2010. When AIMS and Amity University in Noida organized an ICFMT this conference, and he was one of our keynote speakers. A very gentleman, very helpful man. I just wrote a letter to him. I call you on Wednesday. I wrote an email to him Wednesday, and I got a reply on Thursday morning. And he immediately, and the best part was, he said, "I'm now an old man, retired and rested, and I cannot be of much help." That was so beautiful words. Like, he was very honest in saying that although he helped me, he gave me a solution to my problem. But here, I remember I was talking to him regarding the Godra case, which Dr. Bhuvnesh mentioned, because I was facing a narrow polymorphism in that case for the reason because most of the victims were Muslims, and we realized that these family, they were marriage, like these cousins got married within themselves, which is very common in this community. And I had a very narrow polymorphism in this case, and I was just discussing with him. So I had a privilege to meet him. The DNA, that's what Watson says, the molecule is so beautiful. Its glory was reflected on Francis and me. Now the DNA is present in every one of us. And we say that 99% of our DNA is same in all of us. Then what is this DNA expert doing? It is 0.1% of DNA which shows polymorphism and helps us to identify an individual. Not only it helps us to find an individual in forensics, it helps us in the clinical cases, maybe Parkinson, muscular dystrophy, and many other cases. Now in chimerism, because we are working on chimerism, and this DNA profile is a technique which has been used by the scientists to distinguish between the individuals of the same species using their DNA. We have to remember that you have to use the DNA of the same species. You cannot compare humans with any other thing, because you have a DNA in humans, animals, and plants. So you have to have a DNA. You can have to compare the human DNA with the humans, plant DNA with the plants, and that too, not the rice plant with the corn cab or something else. You have to have to try to compare the rice plant with the rice plant only. So such things you have to keep in mind. The traditional method of identification, like see, once the DNA fingerprinting, this technique was established, it was established as I mentioned in 1984. But before that, it's not that our FSL and CFSLs never solved any cases. They had the other conventional techniques to solve these cases. As mentioned by Dr. Bhupender in one of his slides, he mentioned about the fingerprints or the dactylography. Yes, fingerprint or dactylography, as on it, I would consider it as one of the good techniques, but it has its own limitation. If you ask me personally, I have handled, as Dr. Bhuvnesh mentioned, I have handled so many medical legal cases, and most of the medical legal cases I have faced, or the exhibits submitted have been the skeletal remains. So such is the limitation where once I have a skeletal remains, I cannot do the dactylography. So that is the only limitations over there. But yes, fingerprint is one of the good technique. Blood grouping. Blood grouping is again a good technique, but it again has a limitation. See, A positive or B positive is present in most of our population. So with the blood grouping, we can do a screening test. Suppose we have the child and the father not matching, we can exclude it. But suppose the father and the child both are B positive, we cannot include him because B positive is present in 80% of our population. So it becomes a little difficult over there. Protein markers or isozyme markers, those are sensitive markers. When these exhibits are submitted to the FSL or CFSL, it's not that CFSL or FSL, this, once you submit the sample, they start working on it immediately. They have their own backlogs. So by the time they start working on the samples, most of them samples do not work because these markers are very sensitive and the tissues, because India has a variety of season, weather. So they do, even if kept at 4 degrees, they get decomposed and depends upon the preservative. So most of them are preserved in this, not ethanol, they are preserved in formalin. Most of the hospitals, they preserve their tissues in the formalin, and the formalin is I mean, it's not suggestive. There is no other option, but then the samples decompose in the formalin also. So they report either no opinion can be given and something like that. We have the limitation with these conventional techniques like low efficiency, they require different protocol for each locus and low resolution of the band patterns. Then we have the anthropometric analysis also, maybe the superimposition and do the measurement of the but that is again plus minus accuracy. That doesn't give you an accurate identification of an individual depending upon the various factors. But then we have a DNA. We have the advantages. Why do we prefer the DNA? I'm sorry, I'm not here marketing for the DNA. I'm not, but yes, as a scientist, DNA we do prefer in most of the cases now because it has its certain advantages. Is less prone to environmental degradation. As I mentioned that I was talking to Professor Alec Jeffries regarding the Godra case. Trust me, the Godra case happened in 2002 and we came into picture in 2006. That is 4 years past. And even then we got a good DNA, although it has experienced 4 summers, 4 rainy seasons, 4 winters and all that. But even then, after a hard workshop, I believe Bhuvnesh was with me working at AIMS those days. We worked a lot for this case and we got a very good quality, not quantity, only the quality of DNA was very good. Because it's less prone, I don't say it is not prone, but it is less prone to environmental degradation. And the most advantage is that it is present in all nucleated cells. And the best part is it is somatically stable. As I mentioned in my earlier slide, most of the cases I've handled is the skeletal remains. Earlier, the FSL, CFSL people used to get like, we have a skeletal remains. How do we do sexing? How do we do the identification? How do we do this? But now since DNA is there, DNA is somatically stable, which means the pattern of DNA fingerprinting is generated same from the fresh blood, same from the dried blood, same from any part, any tissue of the body. And its uniqueness is it's very unique among the individuals. This is the slide of Professor Alec Jeffries. I have his permission to show you this slide. Professor Alec Jeffries started this experiment long time back in 1884, as I mentioned. And the first experiment he did was for a paternity test, and he used the multi-locus probe. That's a probe which works on all the loci, all the chromosomes, 22 autosomal chromosome and 1 sex chromosome. But the disadvantage here, I would say, is it does not show us the chromosome we are studying. What we do is here, we again work here on the same Mendelian law of inheritance that 50% of the genes from the father and 50% of genes from the mother. So we see the first 2 slides, one is from the father, one is from the mother. We do the statistical analysis and come to the conclusion. Then with the passage of time, the scientists came up with a single locus probe. where we have either one band or the two bands, depending upon the heterozygosity and the homozygosity of an individual. I believe all of you are aware of what is heterozygosity and homozygosity. Fine. So I don't need to explain that. So now the question is, if we have the three bands, it becomes a matter of investigation or a matter of scientific study. It, although not, if not the investigation, yes, it becomes a matter of scientific study. Why do we have the three bands? Because one and two are expected, but the three bands are not, normally not expected. We need to do further research, further study for that. Now, this three generation of DNA profiling, as I said, the multi-locus probe, then we had the single locus probe. Then we had the multi-locus, RFLP contains both a single locus and multi-locus. Then after that, we had the PCR technique. And on the basis of the PCR technique, we used to do HLA DQ alpha. HLA is a gene on the chromosome 6, and we used to do the HLA DQ alpha. It is a PCR-based technique, and we used to do the paternity test. I still remember when I joined CDFD in 1998, we used to do most of the cases using the HLA-DQ alpha. But then with the passage of time, the scientists have come up with the STR markers, and there are certain advantages of this STR markers. STR is the short tandem repeats, and we get a good profile over there. Now, the advantages of this STR is that it is randomly present in our human genome, which makes it very specific and sensitive. Moreover, PCR technique itself is a very sensitive technique. Now, you all have read about the Mendelian law of inheritance, which says 50% of genes from the father and 50% of genes from the mother. But now, I say I have slightly modified this 50% of genes from the biological father and 50% of genes from the biological mother. But what made me to use the word biological in this is that I share a story, I share a case which was related to us by late Professor Lalji Singh when I was working at CDFT. Many of you may have heard about the IVF procedure for the female or the couples who don't have an issue. They go to the IVF centers and for this procedure. In one of the centers at US, there was an IVF center which had a very good reputation because IVFD used to get an success in first attempt. And let me share that it's not necessary to get an success in first attempt. In India, probably we do it for 2 or 3 times, not more than that. Probably 2 times, but I'm not very sure, maybe 3 times, but not more than that. But this center in US, it had a very good success rate, and the females or the couples used to get in success in first attempt. So obviously the customers were quite happy about it. So one day, one of the couple just got curious. They let us find what is this DNA fingerprinting pattern of us and our child. So they went for the DNA fingerprinting test, and the report submitted to them was that the child shares the maternal alleles with the mother, but does not share the paternal alleles with the father. It was very surprising for the father. I don't know, it was surprising, it was insulting, whatever it was for him, but yes, he was not happy with this result. Not that he was not trusting the person who had given the report, but he was not happy about the report. He just talked to a few other couples who had undergone the IVF procedure from this center. When they went for the DNA fingerprinting test, and the same report, the same type of report was submitted to them. So it became a matter of investigation, and it was found during investigation that the doctor who was performing this IVF test used to use his cells, sperm cells, for this test. I don't know how many of you have watched that Vicky Donor movie. Vicky Donor movie of COVID mein dekhi thi. So there the doctor who stores the Vicky's sperm cells for the procedure in his center. The same thing happened here. So I would say that Vicky is the biological father of those children who are born from this center. Or the doctor there in US is the biological father. So this type of case or this type of scenario has made me to add the word biological father and biological mother. Although in COVID, we had another movie, but in that, याद आएगी अभी movie का नाम. Again, they say the DNA of the father does not match with the child, but that was good news. Good news. The movie Good News. But that is not the IVF procedure and all that. That was an IVF case, but not that the sperm cells were exchanged. No. In that case, it can happen by mistake. The sample of one couple was exchanged with another couple. I would say not intentionally, maybe not intentionally, but maybe unintentionally, but this can happen uh, anywhere. But now this technique, DNA fingerprinting technique, I say is a very powerful technique. I don't know how many of you will agree with me. I say it's even powerful than the chair of US president. I don't say any name of the US president. I say the chair of the US president because I have a reason. When I talked about Blue Lewinsky case, Zoom workplace connecting. या तो कोई connect कर रहा होगा. So Blue Lewinsky case, when the ex-president of US, Mr. Bill Clinton, his secretary, she claimed that she accused him or she registered a case against him that she was raped by Mr. Bill Clinton. But yes, then the samples were sent to Alec Jaffrey's lab, where you say Brit is Britain, who's got Bill? Bill is Bill Clinton on the run. Purposefully, the samples were sent to the UK, not to the US for many other reasons. And so it is the DNA technique which examined the undergarments of the Blue Lewinsky and found that the cells of Mr. Bill Clinton were present. So it doesn't matter whether he was a president of US or he was a common man. Yes, but the DNA report was that, yes, he is the one who is a suspect in this case. मैं करता हूँ, ठीक है, ठीक है. अच्छा. Now this DNA profiling technique, it promises correct identification of an individual, except monozygotic twins. I talk a lot about DNA fingerprinting technique. I praise it a lot, and many others also praise a lot about the DNA fingerprinting technique, right? But there are certain novel forensic situations which may prevent the association of the perpetrator of an offense with a stain left at the crime scene or lead to false identification of an individual. Now you look here, this is, one is monozygotic twins, one is dizygotic twins. Can you just recognize the monozygotic twin? नहीं? नहीं, आपने तो सुना है नहीं, आप तो पहचानोगे. Me and my sister.
मैम कौन सी है इनमें से?
मैं कौन सी हूँ, sir? Yes, yes. Me and my sister. My father also had a monozygotic twin brother. Now, the digotic twins, मेरी बुआ की लड़की के बच्चे हैं. उनका एक बेटा और एक बेटी है. Fortunately or unfortunately, हमारे यहाँ continue नहीं हुआ ना मेरा कोई twin हुआ ना मेरी sister का ना मेरे चाचा के बच्चों का कोई हुआ. But yes, मेरी बुआ की लड़की जो है उनके ये digotic twins हुए. Even when we talk on phone, it's very difficult to recognize who is on the phone, is me or she, or it's the same happened with my father. And you won't believe, we stay in the same area, but different sectors. People from her locality, they start talking to me, and people from my locality, they say, and now many of us have started asking, हम 7 सेक्टर वाले से बात कर रहे हैं कि 8 सेक्टर वाले से बात कर रहे हैं. So this, although I say कि अब तो इतनी similarity रही नहीं with the growing age, but still people get confused. When we talked about the somatically stable, that DNA is somatically stable, let me give you an example. There was a case at the scene of crime. We had the blood stain, and we had the 7 suspects in this case. The one of the suspect 5, whose blood sample was taken, and when DNA fingerprinting was demonstrated that his sample or his pattern, DNA fingerprinting pattern, matched with the blood stain which was found at the scene of crime. So this shows it was the multi-locus probe which was used. बहुत पुरानी बात है, around 96, 97 की बात है. Then at that time we used to use the multi-locus probe in India. So what I want to show in this case is does not matter whether you have a fresh blood sample or a dry blood sample. So I have a time limit. Let me come to the now two related new DNA tests, the mitochondrial DNA and the YSTRs, which is very required at times. I'll tell you one. Now, when we talk about the chimerism, why I have started working on the chimerism is that one of the women, she suspected that she has been raped, been sexually assaulted and while unconscious. This is one of the case. The another was, which I read in the paper. I'll not go into details. I have a time constraint. So another is a criminal who was in the jail, not in India, but in somewhere outside India. When he was brought to the hospital for the treatment, the bone marrow transplant, he had undergone the bone marrow transplant. And it was found during this procedure, what he did is he was a very clever man. During his hospitalization, he committed a crime, came back to the hospital, and it took a lot of time for the investigating officers to find out that he has committed the crime, because what he had in his blood sample was the donor DNA. So this is what prompted me to work on the chimerism. I tried to collect the blood sample and different biological samples from the anybody can help me, I cannot see it here. So you can go through this and now you have the two type of chimerism. One is genetic chimerism and one is human-made chimerism. Genetic chimerism is when the two cells are fertilized together, and the human-made is when we have the bone marrow transplant-like situation. So what was very challenging was, in such scenario, what is the best exhibit, biological exhibit, to be submitted for the DNA test? And during my research, we collected the blood sample, hair sample, and buccal swabs, which are very common. And then in my another project, we collected the seminal stains also for the analysis of DNA. But we found that all these three, except hair, shows the chimeric situation from day one, at least till the day 3 months, 90 days, because it was not possible for me to continue my research beyond 90 days because the patient is very difficult to catch hold of the patients. So I continued it for 90 days and I found that here is the only best sample which does not show the donor DNA from the day one, because rest are the tissues and hair is not considered as a tissue. So we don't have a transformation of the DNA into the hair root. It's not the shaft only, even hair roots, we do not have this. Not only that we work on these chimerisms and all that, now presently I am working on the organ transplant also, where as per Form 5 Thota 2014, earlier it is the HLA typing which was used for the matching of the establishing the kinship between the donor and the recipient. But as per these guidelines, the government of India has now now, as per this form, government of India has submitted that it is the only DNA fingerprinting to be used to establish the relationship between the donor and the recipient. So, so we're working on this And earlier, we used to do the DNA fingerprinting on the, say, 2 loci, 4 loci, now 16 loci, but at presently, we're working on 24 and 27 loci. Together, we're increasing the number of markers makes it the statistically more authentic to share the probability of matching and non-matching. And earlier, when we talk about the multi-locus probe or the single locus probe, where we used to have, we required 10 to 15 microgram of DNA to do the analysis, but with this PCR technique or the STR analysis, we require just 1 nanogram of DNA for all these 24 or 27 loci. And a time is not very far off when all the labs will start using the next generation sequencing, where we'll be analyzing 200, 300 markers together. I think I put an end to it because running out of time. If you have any questions. I didn't talk about the cases which I have handled, but yes, maybe next time we can do that. Well, this is a case where one of the army officer was working on the Siachen glacier. He talked to his parents that my duty is over now, I'm going to take rest. After half an hour, the parents get a call that he's no more. And the parents refused to accept this truth. And the doctor performing the autopsy was who was a resident with us earlier at AIM, Dr. Rudra. He decided what to do. He said, okay, Anupama, can you do the DNA fingerprinting test? And the forensic odontology, sorry, not forensic odontology, it was, yes, forensic odontology, the teeth samples were sent. The bones and the teeth were sent. Bones didn't give us a good result because it was all burn, about 80% of burn, because he had gone through a tent which caught fire, and his body, well, he caught fire, his body was burnt around more than 80%. It is the teeth sample which gave us a good result, and we could report that yes, we had his parents. We matched the DNA of the teeth sample with the parents, and we could say yes, he is the sibling of he is the offspring of this couple. So it's helped us a lot. And in the army, in the defense, they take normally 7 years to do all the procedures, but in this case, they didn't have to waste time to really look into it whether it's the same person or not, because DNA fingerprinting report has helped them a lot. So I put in finally end to this. So yes, you have it.
Good morning, ma'am. Ma'am, I really liked your session.
Thank you.
Actually, I had a question that what are the future research area work or the future scope for DNA fingerprinting? Like,
There is lot which I couldn't do in my lifetime, but there is a lot to do.
so ma'am, can you name one or two?
Once you
Which,
start working on it, just let me know.
yes,
I'll give you a very good topic. So I'll definitely help you. Yeah, are you from Amity?
yes, ma'am.
You can talk to Bhuvnesh. Yeah, we
Yes.
come in together. You're doing BSc?
Yes, ma'am.
Yes, so better is that you talk to Bhuvnesh. I'll definitely help you.
Sure, ma'am. Thank you,
But
ma'am.
there's still so much of things to do. Topics like chimerism, it's not possible for everyone. As I said, I have a privilege working in the hospital, getting the sample from the hospital. So I had to do that at least.
Thank you very much, ma'am. Such a nice session.
Thank you, ma'am, for your valuable and insightful presentation. Next, it is my privilege to introduce Dr. Kusum Singhal ma'am, who is joining us online, a research fellow at the University of Aberdeen, UK, specializing in AI-enabled evidence synthesis and data-driven analysis for complex systems. Her work focuses on applying advanced computational approaches, including AI-assisted literature screening, large-scale data analysis, and GIS-based spatial mapping to understand patterns within public health and social systems. With over 35 peer-reviewed publications and active collaborations across academia, government, and community organizations, she contributes significantly to evidence-based policy and decision-making processes. Her work demonstrates how forensic style analytical approaches can enhance transparency, scalability, and real world impact in addressing complex challenges. I request Dr. Kusum Singhal ma'am to kindly join us virtually and grace us with your wisdom. Thank you.
Hi, am I audible? Hello?
Yes, sir.
Okay, thank you. Yeah, first of all, I would like to say thank you to the Amity University and the whole of the team for inviting me. And actually, I was listening Dr. Anupama Rana lecture, and I went back to those days when I approached to ma'am for doing a PhD in forensic medicine because of my background in the health. But yeah, ma'am might have forgotten my name. I was working as a senior research fellow in New Delhi AIMS during that time, and I was quite interested doing the PhD in the forensic odontology. But unfortunately, during that time, getting admission in the AIMS was not a easy cup of tea. So there was no advertisement during that year, and my GRF was expiring. So yeah, thank you, ma'am. Thank you very much for your presentation. And so now, is am I able to share my screen? Okay, so just so I hope this is visible.
Yes, ma'am.
Okay, yeah. So good morning, everyone, and thank you for having me today. So although my session was scheduled a little earlier, but it's fine. So my name is Kusum Singhal. I work as a research fellow at the University of Aberdeen, where we are working on the health determinants is a collaboration, which is a bigger partnership between the Aberdeen city and the NHS Grampian, and there is one another university in the town, Robert Gordon University. So why I have chosen this topic? Because the conference was showing that this gonna be the a sustainable themed based conference, and I'm pretty sure that you have a lot of people who are going to speak about the forensics. So I have chosen a little bit different topic which is of my interest at the moment. So what this means: AI enabled evidence synthesis and the digital innovation for the sustainable data driven system. Because in my current profile we are looking where my job currently sits in the boundary between the academics and the researcher, and working with the local government as well. I'm not just publishing paper and hoping someone reads them. I'm in the room where the council officers and the NHS colleagues trying to make sure that what we find in the evidence and actually what that can shapes the policy. And today, I just want to talk about how artificial intelligence and combined with the right methodological thinking is fundamentally changing how do we do our work. Are the slides moving? Hello? Yes.
We are connecting, ma'am.
Okay. So the session agenda, we're gonna start about the grounding ourselves, like how why this matter and the sheer scale of the evidence problem we are trying to solve. Then we will look at the AI enabled evidence synthesis tool, what they are and how they are like using the we can use in the different terms. So I will start with a question like, how many of you ever been asked by the decision maker, or maybe your line manager, maybe your senior manager, or maybe the senior person, okay, what is the evidence says? And I'm pretty sure that you might be feeling the pressure of knowing the answer. That question probably could take more than month, because generating evidence is not an easy task, whether it is in forensic, whether it is in public health, whether it is in any kind of sector. That gap between the question being asked and the evidence being available is what we are trying to do here. Yeah. So let me paint a picture where we can talk about the what are the challenges and the landscape. Definitely, whenever someone is asked to do some kind of evidence synthesis, then the biggest challenge is the volume and the velocity. There are over like 2.5 million studies published annually, and this number keeps on increasing, especially after the post-COVID era, because during that time there was no specific lab works was happening in any of the lab because of the COVID situations.
Ma'am, can you
And
just
what
take a bit pause because we are just some settings has been changing, so they are just doing on ground, so we just inform you.
Okay, sorry. Thank
Now you can.
Yes, ma'am, you may start the session now.
Okay, thank you. So now,
And ma'am, if it's possible, just open your
sorry.
camera and can you just move second slides also, not back slides, two back slides.
Yeah. Am I visible now?
Yes, ma'am, you are visible now.
Okay, thank you.
Just ma'am, start from the
So,
two back slides.
so you this one?
Yeah, ma'am, you can start.
Okay. So this is the overview of the presentation I'm going to talk about today, that what are the challenges, why the AI enabled synthesis required, why we are talking about the large scale evidence mapping and the digital innovation methods, along with the sustainability and the policy impact, and then the last, definitely the conclusion and the question answers. Yeah, so this here comes the challenges. So like uh why AI enabled evidence synthesis came into so much limelight. What were the challenges we were facing before that? Because whenever someone asks you to generate some kind of evidence, that needs a lot of work depending upon the topic which you are looking through. So for example, if you want to do some kind of evidence informed decision making, the first challenge is the major one, that is the volume and the velocity. And there are over 2.5 million papers which has been publishing every single year, and that number is not slowing down, that is now increasing, because every single step, for example, if we talk about, okay, I want to start working on some of the topic. First of all, I need to look into the literature, where is the evidence, whether this kind of interventions is really required or not. Secondly, definitely the complexity of the evidence. Here, as at Aberdeen, we are often dealing with the problems that came across health, social care, and the environmental problems. And thirdly, the policy urgency. This, I think, this is the one of the critical situation we are now, because government always keep on asking the rapid and high quality evidence to inform the legislation. They cannot give you 10 months or like 12 months to sit on one topic and then keep on thinking, working on that. So they want the result very quickly. I think that's why AI is here to solve this purpose. Then keep coming the equity and the inclusion. This one is close to my heart. In my work, we have been doing the work on the food insecurity and the other social issues like asylum seeker health, because the things now has been changed because of this all the Ukraine situation and the war situation. So they want to include every single person in the policy. So that's why equity and inclusion is also a major challenge. And the reproducibility crisis. So many policy decisions rest on a fragile kind of basis, because there are not enough studies on that topic, and especially there is a limit for the replication as well. And the last but not the least is the digital divide. This tool I'm going to show you today, like, I think this is the powerful one, but they are not universal accessible. If we are not careful, we build systems that work brilliantly for well-structured institution and leave everyone behind. So my purpose of putting this slide here, before jumping into the AI enabled evidence synthesis, we really want to know what are the challenge, why we are talking about the AI based things everywhere. So I think this is the more of the last slide. I already mentioned that 80% of, so for example, you start doing some research in on topic. I think the most 80% of the time of any of the reviewer will go just in screening. So I still remember the days when I started doing my PhD. So I think the first initial 6 months I was just doing the literature search, going through the papers, their PDF, sitting in the lab, keep on reading the papers before jumping on any of the conclusion that okay, this is going to be my topic. Here there is a gap in the literature, this I can start with. So it takes a lot of time, and this is not fast enough for the world we live in. This is a very bitter truth, because nobody has that time to give you 1 or 2 years, or no, none of the supervisor or none of the line manager or the policy makers give you that much time. So now the good news is, with the AI assisted screening, we can achieve 30 to 40% reduction in the cost, especially cost in case of time, not in the monetary. I'm saying sensitivity means we do not miss any of the relevant studies, can reach 95% of the, if we are using the proper model and we are giving them the proper prompt in form of the so I think all these large language models, LLM we call it in a short form, they are actually reducing your time from, like, any time from 3 to 6 times. They are speed up your procedure. So yeah, so it's not like that AI is completely new. It's not. So why this conversation is happening now, why not 10 years ago? The answer is that several things converged at roughly the same time. The transformer architecture in 2017, think of that, that the technical big breakthrough made have been made through the AI learning models. And in 2022 we had a domain specific models trained specifically on the scientific literature. And by the 2022 we all have been aware, by the chat GPT something shifted in the public consciousness. Suddenly everyone, including policymakers and the council officers and any of the local government, had the person experience of what AI can do. And in our work, we are most of the time we are using the systems like EasySLR and RAN. So this time these names might seem fancy to you, but if you do the Google, they are quite, like, they are a plethora of systematic review models are already there, apps are already there, AI based softwares are already there. So you really have to look into that which tool is going to solve your purpose. And nobody can tell you in this world that, oh, this is going to solve your purpose. It depends really what you are looking through. If you want policy based evidence in with a high quality, then they are different. If you want something just to do the screening and searching, then there are different tools. So it really depend upon your choice and the time limit you are having. So this is the methodological core what we do. I want to be specific because there is a lot of hype around the AI around nowadays, and I think it does more harm than the good when people are not precise about what these tools actually do. Yeah, so this is there comes the concern. You have to be very precise about these tools. So I think this is the simplest definition I can put here. AI enabled evidence synthesis refer to the application of all these natural language processing and the computational methods to systematically identify, screen, extract, and synthesize. I want to be clear about something. AI cannot replace any of the human judgment in the process, whether you consider any single domain, whether it is physical sciences, biological sciences. AI cannot replace the human judgment in this process. It can amplify. So there's a difference between replacement and the amplification. It can amplify the human capacity, and it still makes intelligent decision most of the time has to be proper human intervention, proper human calibration that need to be always, there should always be a supervision of the human while dealing with these AI softwares. So I think I still make the call about what evidence means for policy, what AI does in handle the mechanical, repetitive parts, reading hundreds of thousands of abstract and extracting data from them, so that my time and my expertise now is going towards the effective part, which really needs the human brain, rather than the part like going through abstract or just reading a small paragraph or reading a 20-page report. So I'm not devoting my time towards that. I'm just putting them in the AI softwares and do the summary and get to know that, okay, this was the thing. So this actually saves a lot of my time, and most of my expertise and skill goes towards the things which really needs the attention rather than just reading through the literature. Yeah, so this is actually the workflow. I think most of you might be aware, because evidence synthesis is, it's not new in India. Also, a lot of people are doing evidence synthesis. I think after COVID, people got their interest more in this, because there are like thousands and thousands of published literature just on single topic, and we don't know which paper is having a good quality, which paper is having the authentic data. So I think people, that was the motivation of doing the evidence synthesis. People started actually looking into the evidence, where is the evidence, whether this is required or not required. So the system starts with the searching and retrieval. A lot of databases are there, for example, Medline, PsycInfo, Embase, Gray Literature. It really depends upon the topic you choose. For example, if you are working on some of the topic of the mental health, you need to check into the PsycInfo the database. If you are working on a medical topic, then Medline would be the great one. And after that, we jump on to the deduplication of the records and import them into the screening tool, then AI pre-screening tool. So I think this is really important, because these machine learning models reads each title and abstract and assign a probability score, how likely this study can be relevant to your work. High scoring reports goes into a human review. Definitely, I'm saying again and again, AI cannot replace human judgment or human surveillance. Human should be always there. So but AI can little decrease your you know uh, time devotion and on which topics you really have to look upon. Then there comes the full review. After having the human calibration, there comes the full text review, and definitely that steps goes into the data extraction, because the after all, why you are looking into that paper, because you want to extract some kind of data, you want to know what the paper was saying and in what, on which population the paper was based. So because AI has narrowed down the candidate pool, the work is more focused and efficient now, which is in great sense. I think that can be used for the human welfare, and at the end, synthesis and reporting and the evidence maps. So these are the large language models in the evidence synthesis. I just want to address very quickly. So the traditional manual screening runs about 100 to 200 per hour, because I don't think so any of us can do more than this screening. 200 reading, 200 abstract in 1 hour. It's a huge, but AI model can read up to 10,000. So that's the biggest difference. Then the consistency. So that's why the training world came into the picture. These AI models needs training, consistent training. They to build their databases, to repeat their result. Because if there is no repetition or reproducibility in the result, then that is a big question, that why there is a discrepancy. But if you talk about the human, if human gonna do so, for example, even if you talk about the forensic, I cannot write my name in the same manner if I write it again, because that's a normal human enter rate variability. And but AI is just machine that will reproduce the same kind of results. If you train the model again and again, they will so there is no doubt about it. If you have your inclusion criteria, if you have something in your mind, okay, I want something on specifically this topic, that will produce specific the same results again and again. But I cannot stress this enough, that speed advantage only counts if we maintain the quality. Yeah, so So that's the thing people don't understand. We are living in this era of AI nowadays, that we are ignoring the quality. So I think speed matters, but with the speed, I think the most important thing is the quality. The row of consistency matter. Human reviewer, even experienced one, disagree with each other after 14 to 20% of the time. I think the same thing I'm saying, like being a human, if I write the same word, my right handwriting would not be same. There is not like even a 1% probability that it's going to be exactly same. It cannot be. So in that context, AI can really help you. And it's always a good thing, not necessarily. A model trained on the biased data will make biased decision. So that's why I'm saying that proper training of these AI models is really important, because if input is not okay, you will always get. So there is a statement, I think, in the science, if there you put garbage in, you will get the garbage out. So there is no point of discussion on this thing. So human oversight of a model performance, definitely this is required. So this is a very brief of which I already discussed, that this was the initial search, then AI screened this thing, and we just got only 6 weeks to complete this evidence synthesis. So in that sense, AI is really brilliant. There is no question about this. Yeah, so now I'm going to talk about the large scale evidence mapping, which consists of GIS, spatial analytics, and the pattern identification at a scale. So an evidence map is essentially a systematic catalog of the research landscape on a given topic. Rather than synthesizing findings, which is what a traditional review can do, it asks what research already exists, where it is concentrated, and where are the gaps. That's we want to know. That's why we are doing the evidence mapping. So like we have done a couple of different scopy review, and all of them are related to the public health. So by doing those reviews, we really can understand, okay, there was a lack of evidence, there is the proper intervention required. And here comes the most of the agencies, like if we talk about the government agencies in India also, they are really keen to know that, okay, where we have to invest our money, which is the deprived population, which really needs our help. So I think in this context, you by using these models, that will decrease your time and increase your efficiency, and you can just provide the report, okay, this is the area where we can just focus on at the moment. Because all these country setups, definitely they don't have the biggest amount of the budget to put into the different domains. So they always assign some kind of budget, and they have so they are very like peculiar to put, okay, this is the important, this is on the priority scale. So the bibliometric analysis and the scoping reviews are the methodological tools, but the special piece, the GIS piece, is what makes the evidence visible. In what way? That genuinely intuitive for the non-academic audience. So because academician, so I was working in the university before that also, but being academician, we keep on generating the paper, and our audience, we they don't have any idea what was the results, because they are not the academician, they are not here to read the whole of the paper. In that context, like GIS piece of work really helpful, because that can help you. So GIS is a geographic information system. They can really help portraying that, okay, here is the gap, here is the work, because every geography is different. So the fundamental insight in this research has a geography. Studies are conducted in the particular place with the particular population in a particular context. So that's why when we start working, our PhD supervisor used to ask, oh, which gonna be your population, in which gonna be your area. So my PhD was related to the Haryana population, and the definitely the geography was Haryana only. That's a state in north of India. So when we map where is the evidence, where it is not, we actually reveal things that a traditional narrative synthesis would never do, right? Because narrative synthesis will is not done in a systematic way. We just read, I think 20 or 30 or 50 paper depending upon your topic, and we just generate a report. That's the literature, that's the narrative synthesis. But that's not a systematic review. That's not that you cannot generate the evidence, because you haven't searched all the databases. where the evidence already existed. You haven't searched any of the gray literature. You haven't searched any of the websites, like some of the websites, for example, ICMR has their own website. They sometimes they put the reports on their website. The paper, if they are not the academic paper, you cannot find them on the Medline, PubMed. You cannot find them. You have to go on their specific website and to try to find. So, but beauty of this thing, evidence synthesis, because they are done in a very systematic and robust manner. So there is no doubt about it that they're gonna capture every single information on that topic. So why spatial analytics lets us to ask? So because there are specific areas where, for example, if I talk about my work, so areas of Aberdeen which are having the worst housing condition, because I think most of these European countries have this issue. They are very cold and people are struggling with their, because they don't have that much money to pay their electricity bills because of the inflation rate. It's so are those are the same areas that are the underserved people also, and they really need required to be required there should be intervention. Are those are the area where the research evidence is thinnest. So when those three maps overlap, then this is we called where we should direct the intervention. For example, like we found, okay, there is a poor housing, there is a poor health, and there is a poor evidence as well. So when these three things comes in the picture, then this is the place where the policymaker should intervene. They should make some policy to direct their budgets or funds in that direction. And this picture can be generated only and only with the help of GIS and the spatial analytics, rather than just writing a paper. So yeah, so this is the chart which showing something I think I find striking. Look at the distribution of the evidence across the social domain: environment, employment, social care, housing, child poverty, education, and the mental health. So definitely the mental health is the biggest one at the moment, because after COVID situation has been changed, people are struggling because they are not having their social conversations. They are living in the isolation. So I think this is the second last component of the digital innovation methods. I really want to zoom out here that broader ecosystem in which these tools really sits, because AI and GIS does not work in isolation. They work as a part of interconnected set of methods and discipline. So think of this hub and, like, for example, the systematic review process, the rigorous methodology, and the feeding into these different directions, like natural learning program, and then mining, machine learning, they all are equally distributing in this evidence synthesis hub. What makes this powerful is the natural learning processing extract structured data, and then machine learning will help generating the evidence, and then with the help of GIS and spatial analytics, we can actually show pictorially, which also leads up to the data visualization, and then open research, and then this information goes to the policy informatics. So this is quite a cycle, which is, I think, quite good to provide the evidence. Yes, this is the thing, interdisciplinary methods in practice. Definitely, none of the science exists alone. This is the interdisciplinary work. My background in the public health and then dental science, along with the systematic review methodology. So, and my colleagues are working in the different domain. I cannot work because I'm not a statistician. I cannot run all this work. I'm not a geographical person. My background is, I don't have any expertise about the geography. So this work I'm talking about, and definitely not a IT person, not a computer science person. So I don't know. I can run the machine learning software, but I need someone to work on the background who is having this kind of expertise. So what we have found generally, not just the theoretically, is that we must interesting insight came from the collision between different perspective. For example, when a statistician and a social worker look at the same data set, they might be having their different kind of vision, because one is statistician, one is the social worker. When a geographer and the public health researcher look at the same time, they have the different type of question. They will look the research from the different angle because of their background and their expertise. So that's why the change of the course is the true interdisciplinary work is hard to find, hard to publish, and hard to evaluate. The academic incentive structures were not designed like this. So yes, I think I'm just running out of time. Yeah, so this is the data quality and governance and ethics, because I mentioned in the previous slide also, human surveillance and human calibration cannot be compromised at any cost, because that will definitely going to hamper the quality. So human oversight, mandatory human review at the inclusion and exclusion decision important. And any kind of research, for example, I'm working in the UK at the moment, so there are different kind of compliance. If you are working with the clinical data sets, if you are working with the non-clinical data sets, so you need to get some kind of training. I think every country has their own setup of training. So UK is not UK is not that much fancy, like they are doing something unique. No, every country has their own. So I think in India also, when we start doing any kind of research, we need to go through the human ethics. If they are working on the animals, then there are different ethic boards from the on the university, or all the medical institutes are having. So these kind of compliance to those principles of those is really very important. Sustainability. So I think this word comes, I think, in every domain. Even the government keep on talking about we need the sustainable solution of blah blah blah. So sustainability is the biggest thing. We already talked about the methods, tools, and now we want to talk about what all of these actually for. Because the test of any research system isn't whether it's methodological elegant, it's whether it makes a single things better for the people. Sustainability, so there are different kind of sustainability. Technical sustainability is there, organizational sustainability there, knowledge and environmental sustainability there. I'm not going into detail because it's a yeah. So evidence to policy impact. So this impact pathway that I care most about, I want to walk you through with a very quick example. So we have conducted a review. In case, for example, we have done the warm house prescription pilot evaluation. So we use the data from the Grampian Data Safe Haven. That's a one kind of organization. So to identify fuel power household eligible for the minimal support, then evaluate the health outcomes of those and who received the intervention. Aberdeen accounted for the majority of the pilot participants. So I think in the policy dialogue, those outputs definitely are shared with the city council and the Grampian, and entered into the national cost of living policy conversation in Scotland. Decision and action. The findings have been cited in the national policy discussions about the financial support, and then this will think that evaluation and the learning should be important. For example, any of the policy maker, they have implemented any kind of intervention, but if it's not evaluated, then there is no point of doing this, because we really want to know why the intervention was done and what was the impact of that. So these are the challenges and limitations. I think most of them I have already talked about, like, for example, model bias. So most of the serious issue, I think, in my opinion, because AI system are trained on the biased literature, then it will always and always produce the biased. So these models needs a lot of training. I think most of the chat bots are very rubbish in that context, because they are, they are just chat bot. For example, chat GPT, they can just help you to give some direction, but they are not the right platform to do is. So, and definitely where comes the computational codes. Large scale interference requires a significant resources, because the people, why they are not using. So the I think my thinking behind this, because I'm working in an organization who have the subscription for 2 or 3 of these AI tools. But if you are not having those kind of budget, then it's a problem. Then tool fragmentation, hallucination risk, skill gaps, and the evaluation deficit. Okay, so the future direction and the emerging opportunity. So these are the multimodal evidence synthesis, integrating not just about the text, but tables, graphs, figure, images, and eventually audios and videos data sets also. Because that's what I was saying. Nowadays, we don't need data just in a publication form. We need data to be more visually appealing. We need data in the form of graphs, so that with the graph only, they know that, okay, this problem needs some kind of intervention. This problem does not need any kind of intervention. So that's why in that direction, these models can be trained. And global south equity, this is one that I think about most honestly. The tools we are building reflect the context we are working. If those tools are going to be useful globally in low and middle income countries, in under-resourced health systems, that has to be a design requirement from the start, not after that. So recommendations and the policy research teams adopt active learning screening tools. They are good. The funders, so these are two funders, UKRI, the and the NIHR, National Institute of Health and Research. It's I think corresponding the same. We are having the ICMR in India. The policy teams, university develop the interdisciplinary training program. So that I think the people are really hesitant about the collaborating things. So I think there should be proper interdisciplinary collaboration and training programs to learn all these tools. Not all this, maybe one or two of them are sufficient enough, they are, if they are good enough and they are trained properly. And then technology teams, so priorities open source interoperable tools. Nowadays a lot of like open science framework is there, open science is there, NIHR open science is another one where you can just put, rather than properly publishing the paper. So yeah. Okay. Oh, sorry, I just rushed in the last 5 minutes. The conclusion and the takeaways. So five things I hope you will leave here after listening this presentation. AI amplifies, not replaces human expertise. So don't worry about that. Nobody is going to take our job. No one can take our job. Every AI assisted review I have led is required more human judgment, not less, just applied in the different places. Secondly, the scale demands new methods. We are in world where the traditional approaches can keep up 2.5 million papers per year. So yeah, that's the biggest worry. Spatial thinking matters. The geography of evidence is real and it's unequal. When we map where the research is, where is not, for sometimes we keep on coming off the paper, oh, this study is just happened in the African countries, none of the studies happened in the Europe, none of the study happened in India. So I think the geography and spatial location of the evidence is really very important. Interdisciplinary is the foundation, no question about that. The transparency build the trust, because the AI models are quite transparent. They are not like, okay, I'm doing this thing, if my friend wrote that paper, okay, that's going to be good quality. No, they are very transparent. So that's a good thing about those. Oh, thank you. Sorry. Yeah, so it was a rush in the end.
Thank you, ma'am, for that engaging and enlightening session. I would now like to once again request our esteemed dignitaries to please step onto the stage for the felicitation ceremony. Does the audience have any questions for ma'am? No, so I would like to invite our esteemed dignitaries to please step onto the stage for the felicitation ceremony for Dr. Amarnath Mishra.
Thank you very much, ma'am. It was a nice session. Thank you.
Thank you.
हाँ, मैं भी सुन रही हूँ।
It is a great honor to introduce Professor Dr. Amarnath Mishra sir, an eminent forensic scientist, academician, and globally recognized research leader. Currently serving as the Associate Professor at the MIT Institute of Forensic Sciences, he brings over 18 years of extensive experience in forensic research, teaching, and medical legal consultancy. His scholarly contributions include over 120 research publications, numerous book chapters, authored books, and several patents. Recognized among the world's top 2 scientists in 2025 by the Stanford University and Elsevier, Dr. Mishra has received multiple prestigious national and international awards. He has mentored hundreds of students and trained professionals from agencies such as the CBI and state police forces, significantly contributing to capacity building in forensic sciences. I now invite Professor Dr. Amarnath Mishra sir to grace the stage.
Good afternoon, all of you. Thank you, organizing team, for inviting me. And now I want to start my talk on my topic, global challenges and innovations in forensic toxicology. So as today this conference is interdisciplinary and where forensic science is also one of the interdisciplinary sciences. Application of science and technology in investigation and detection of crime and administration of justice. So one of the subject is forensic toxicology, that is very specialized and also linked with basic science as well as forensic medicine department, because there is dedicated lab and facility for toxicological interpretation, analysis, and more. When we are moving on this topic, first of all, we are looking for Paracelsus. Any substance may be poisonous. A good medicine in large doses may be poisonous, and a poison in small doses may be a good medicine. Only the dose does, the right dose differentiate a poison and a remedy. So when we go for the definition of poison, any substance in the form of solid, liquid, or gas, when administered in the body, that cause deleterious effect. Okay? So that is very.
Sir, your slides are not moving online, so just kindly just reshare because we are on a, right now we're on our first slide.
Yeah, sure. No issue.
Just stop share and just reshare.
Is it visible now?
Yeah, it's visible, but it's not a presentation mode. Just put in presentation mode. No, it's not. Do one thing, na, just stop share and just choose the whole screen. Do not choose the PPT only. Now go on Zoom. Press escape, na? Press escape. Go to display settings. Are you able to see now? Top display settings.
So now it's visible.
It's visible, but you have it's coming on a dual screen mode.
No, sir.
Are you able to see? There is an option on display settings. And top there is a display settings, na?
Is it visible now? Is it moving?
No, ma'am, it's not. Yeah, it's moving right now.
Full screen.
So now it is moving.
Yes, sir, it's moving, right?
So
Yeah,
let-
it's moving, sir. Moving. Thank you so much. Moving.
So we know that on the basis of the dose, okay, like you already, many students are scattered, not sitting in front row, okay. So I hope you have proper energy right now to listen, yes? Okay, very good. So that depends on the dose because many lectures already you have listened, okay? So don't mix, all are separate and that is application of forensic science, that is science and technology, okay? So Paracelsus talk about dose, okay? Then next, Matthew Joseph Orfila make chemical analysis a routine part of forensic medicine and made studies of aphoxisation, the decomposition of bodies and exhumation. So what happened in cases of forensic science and forensic medicine? Okay, generally, you know what is the autopsy, what is the forensic medicine, that is application of medical knowledge in administration of justice. Okay, so when dead body suspected in suspected cases, okay, if that is a unnatural death and we don't know cause of death, generally when that body come in the mortuary house, we go for the examination for cause of death, mode of death, manner of death, and also mechanism of death. Okay. So in cases of poisoning, we required to collect the viscera, okay, and send to FSL for further analysis, okay. So without knowing about which poisons, we are unable to tell about if that case related to suspected poisoning. So that is very important, and Varifla first go for the chemical analysis part of Visra, and he is also known as father of forensic toxicology. Okay, one of the foremost Indian expert in the science was Kashyap, a physician who lived in the times of Buddha, that is 6th century BC. He was a successful curer of snake bite. So, in India or in Nepal or in developing country, that is good forest and there is a chances of snake bite. Okay, so he also a well-known physician that time in curing of snake bite cases. Now we are talking about Dr. Sibte Hassan Jaidi. Dr. Jaidi is the best known for his experiments that describe the effects of silica, coal dust, asbestos, synthetic dyes, and heavy metals on the human body and their role in diseases such as silicosis and coal workers' pneumoconiosis. He was also instrumental in founding several institutions related to industrial toxicology and occupational health in the Southeast Asian region. So one of the organization, that is Indian Institute of Toxicological Research is Lucknow, that is CSR lab. For his contribution to the development of the specialty of toxicology in India, he has been often credited as the father of Indian toxicology. Okay, father of Indian toxicology, just Dr. Lalji Singh is father of Indian DNA fingerprinting. Okay. So you know the terminology of toxicology, forensic toxicology, and already I told about poisons. Okay. So when we are talking about toxicology so in whole, that is study of how natural and poison cause desirable effect in living organism. When we are talking about forensic toxicology, that is the multidisciplinary field of involving the detection and interpretation of presence of drugs and other potentially toxic compounds in bodily tissues and fluids. These analysis and interpretation are conducted in a manner to differentiable in court. So what happened in the forensic science laboratory as well as toxicology lab in any medical, in like tertiary care hospitals, tertiary care hospital, there is also toxicology unit who interpret cases of poisoning, and that is very essential to present in the court of law to know the actual cause of death if that is case of poisoning. Because in many cases, like suppose it is hanging. So in hanging, what happened that is antemortem or postmortem. So suppose if somebody is given some drugs or poisons before hanging, so death occurs due to poisons, then that body is hanged. So we can go for the postmortem. Hanging means that a person killed by some another person. That is not suicidal hanging. Okay, so we can interpret such type of things on the basis of the interpretation of poison. So a drug is a substance that is used or proposed to be used to modify or expose physiological structure of pathological states for the benefit of the recipients. So this is the drug, and that then goes for overdose of the drugs, like in cases of uh, so what happened? Like in cases of many cases that is related to poisoning, like Sunanda Pushkar case, or that is many times due to love failure or failure in the examination, adolescent or adults taking such type of medicine in multiple times, like overdose of paracetamol or any psychoactive substances like sleeping pills. So such type of cases happen. So in that conditions, our duty is to find the truth, what is the actual cause of death in such type of cases, and that is only on the basis of the analysis of that viscera or that tissues. Hardly a week goes by without hearing that chemical potentially threatening our health, like pesticides in the food we eat, pesticides. So generally there is uh, exposure of pesticides in crops is also a major threat, and issues related to accumulation of such type of thing, the body as well as also a standard form in the form of cancer and other thing. Last time you listened the um, adolescent adulteration in cases of some milk, like different brand like Mother Dairy, Amul, and other. So what happened routinely? Quality check is required about the food products or drinks. That is very important, and then recommend for the eat. Okay, pollutants in the air we breathe. Now, we also there is air is polluted during the time of Diwali, so many people go for the air purifier. Okay, because proper air is also required, proper water is also required. That is not huge TDS, that is less than 150, 300 for the proper drinking water. So such type of things is very important. And if you are taking polluted water and you are inhaling such type of air, so that is very dangerous for the health. Chemicals in the water we drink, toxic dump sites near our homes. So there is also cases of uh, in, um, uh, that is exposed in your crops, your water, or your usable things. So which chemicals are really dangerous? How much does it take to cause harm? What are the effects of a particular chemical, cancer, nervous system damage, and birth defect? So, you know, the exposure of during the Second World War, there is atom bomb was there in Japan, and exposure was there. So, due to chemical exposure, that is impact in the gene level. So, there is a birth defect. Okay, in a poisoning cases also, if there is a proper exposure in like industrial area, if people is there are exposing of water and food products, so that is also very harmful. Um, and that chemicals, suppose like we are taking coffee, that is cancerous things, that caffeine is in a part of ppm, ppb, okay, ppb. So what happened, that is going into the body, but not in such amounts that cause the ill effect. But when we are taking more exposure, so there is a chances of disease like cancer and other skin issues. Now, the responsibility of the toxicologist is to develop new and better ways to determine the potential harmful effect of chemical and physical agents and the amount that will cause these effects. Design and carry out carefully controlled studies of specific chemicals of social and economic importance to determine the condition under which they can be used safely. Assess the probability that particular chemicals processes present a significant risk to human health and the environment, and assist in the establishment of the rules and regulations aimed at protecting and preserving human health and the environment. So there are water board, there is also agriculture scientists, and many peoples also go for the routine checkup, just like we are go for the routine checkup of our health. So water of different areas, if I'm talking about Bathinda, I'm talking about Balia in Uttar Pradesh. So there is water is not proper drinkable, and there is exposure of heavy metals. So proper quality check is required that which type of water we can take or not. In Delhi, many people are taking Bisleri water in the bottle or Aquafina, different brand water. So what happened? Maybe there is also chances of that water is infected. Okay, maybe that is having the TDS, because if that is not quality check at the in the factory, that is also a big issue. And what happened, that is in the plastic can, okay, plastic cover. So there is also exposure of microplastics, okay, microplastics that is very harmful. Like I'm going to start my vehicle, so just I open the glass, then start AC, then close. So there is a chances of microplastic. We are taking, if using plastic, any plastic things we are using, like potato chips in the Lay's packet or anything. So during process, after preparing packaging and forwarded to the different shops. So what happened? There is a proper exposure from the heat and other things, and there is the deposition of microplastics on that every chip. So everything is dangerous if you are taking in the large amount. Our junk food and packet food always not always that is lethal, that is harmful, but people taken due to some reason or due to not proper handmade food. The responsibility of the forensic toxicologist is to forensic toxicologist work in a lab to identify poisons. Okay, so that lab, academic lab for the purpose of academic regions for your practicing, okay? But for the court of law, you required proper lab setup with the scope of that lab, like preliminary examination and confirmatory test to deposit the report in the court of law, okay? So that lab can also establish in tertiary care hospitals like AIMS having a toxicology unit, AIMS New Delhi, AIMS Deoghar is establishing, and also in AIMS Gorakhpur is also starting such type of activity. So what happens? There are plenty of cases of poisoning. So when we are talking about central forensic science laboratories or state forensic science laboratories or new labs, so what happened? There are plenty of cases, number of cases. So we required more labs to solve that analyze that samples in proper time durations, and that will also help in cases that will not take more trial dates. Okay. The forensic toxicologist needs to work patiently to gain reliable identification using chain of custody for physical evidence and clearly documenting every step taken. When I first time visited a forensic lab in Hyderabad, so I asked with one forensic toxicologist, sir, why you are taking a sample of urine and blood of railway workers? That it can also examine in any pathology lab. So thing is that two things very important in forensics. First, that is related to court of law and that is legal things. So we requires proper integrity, proper securing that evidences and proper chain of custody and reporting. That is very important because on that basis, you decide fate of a person, future of a person. You can also punish or you can also, there is no punishment on the basis of the report. Second, in forensic science, samples are very challenging in trace amounts. So you don't have multiple time to examine same samples. So you required trained people, trained experts, okay, and trained and good sophisticated instrumentations for proper and accurate results. That is very important, okay. So the forensic toxicologist must be ready to explain their conclusions clearly and in details to a jury and to defend their results under cross-examination in the courtroom. Generally, what happens when you are examining, like you are using any preliminary examination or any confirmatory test, so that is based on the methods. Okay. So in forensic toxicology, generally they ask about results, process, not in deep. Generally, like I am also an individual technical assessor of chemical sciences, so they not describe every methods, okay? Just he refer, this is the particular test, we examine with the chemical examinations and this is the results, okay? There is the proper manual, proper SOP in the particular laboratories to examine such type of cases, okay? So that is already SOPs, so there is no challenge on the methodology and already that is the accredited lab, people are come from the proper test and exams, so that is trained peoples, knowledgeable peoples, okay? And if required, they also go for the training during the probation period. So they must also be able to provide an educated opinion on what effect a given substance would have on an individual. Now, global challenges in forensic toxicology. When we are talking about challenges in India, limited resources and training, forensic labs often lack modern technology and trained toxicologists. There is a need for continuous professional development and certification. So already I told that there are different FSLs which also having the forensic chemistry and toxicology lab for the examination, and medical colleges like AIMS, okay, or PGI that also having the toxicology lab that also going to reduce the number of pendency, okay. So, and also such type of lab is developing on daily basis, on annual basis. There is a proper plan of the Ministry of Home Affairs. There is a need for continuous professional development. So for the faculties, there is a faculty development programs, and also good CSIR labs and other laboratories offer training, professional training for new development and new advancement in the field of forensic toxicology that will help to reduce the limited resources and training. Backlog of cases, high case volume leads to delay in toxicology report, especially in rural areas with logistic challenges. So if there is a pendency in the toxicology cases, so what happened? Problem is that Visra required proper preserving things. There is a proper refrigeration is required for not decaying of that visceral samples. Okay, so storing is the big task in laboratory, which is in not in urban area, okay, suburban areas. So proper electricity supply is sometimes fluctuate. So there is a chances of damage of that sample, that exhibits. So such type challenges can reduce on the basis of solving or analyzing such type of cases on priority basis, and there is the proper time durations is required. Then unregulated use of chemicals. Widespread use of unregulated pesticides and chemicals causes frequent poisoning incidents, complicating death investigation. Legal and regulatory issues, inconsistent forensic protocols across states lead to testing discrepancies, and there's delay in implementing updated guidelines for emerging toxin. Public awareness and training, limited public awareness of toxic risks, particularly in rural areas, and insufficient training for law enforcement and health workers on handling toxic substances. When we are talking about poisoning trends in United States, opioid crisis is there, fentanyl and heroin, such type of substances. Still research are going on such type of topics, okay? Household products poisoning from cleaning agents, pesticides and drugs. Then environmental concerns, exposure to lead, arsenic and industrial chemicals. So industrial exposure is also used talks. Also, we are studying in environmental toxicology for the purpose of environment, and also that is unintentional poisonings, okay, that is not suicidal or homicidal in cases of forensic science. European Union pesticides and agrochemicals, common cause of poisoning, particularly in agricultural region. Pharmaceuticals, prescription drugs misuse, okay, prescription drugs. So there is a abuse as well as addiction of such type of drugs is also very harmful for the health. And again, this is not suicidal or homicidal poisoning, that is accidental or due to unintentional. Lead poisoning, children in Eastern Europe will still affected by lead exposure in older housing. Africa, in ingestion of household chemicals, high rates of poisoning from cleaning agents and other chemicals. Lead exposure from informal mining, especially in West Africa. Snake bites are significant cause of poisoning in rural areas due to a lack of antivenom availability. China, industrial toxins, high exposure to industrial chemicals and heavy metals. Air pollution leading to respiratory and cardiovascular toxicology issue. Pesticide poisonings, accidental poisoning due to agriculture chemicals. In India, Pesticide poisonings, high rates of pesticides related poisoning in rural areas. Drug overdose, increasing number of cases related to misuse of pharmaceutical drugs. Heavy metals and fluoride poisoning due to contaminated water, food and herbal poisoning, toxicity from unsafe food practices, and the misuse of traditional herbs. So already many scholars, students, and peoples are working in the pesticides poisoning. Overdoses cases is also reported, and there are many misuse of the drugs, like in cases of date rape cases, okay, in cases of stupefying, in cases of any such type of theft cases, also giving some psychoactive substances, okay. Then water contaminations. Suppose if our waste things from the houses goes in the water, there is also exposure in the water of that different types of drugs. That is also many studies are going on in cases of water contamination, and waste products from the hospitals are from the drugs. Food and herbal poisoning is still the part of FSSI, and also if that is a case of referred to forensic lab, we analyze such type of products and identify the chemicals. When I'm talking about Nepal, my personal study of doctorate is in Nepal poisoning trends. So there is also pesticide related poisoning, drug overdoses. So main is pesticide poisoning due to agriculture rich or green belt is there more, and also snake invasion cases is reported. So this is during my Nepal study. I also attached with Center Forensic Science Laboratory, Lalitpur, okay. Some papers related to medical legal organophosphorus poisoning in central region of Nepal, some books. Now, global challenges in forensic toxicology. Complexity of emerging drugs, like rapid development and modification of designer drugs to evade legal regulations. Okay? This creates challenges for forensic detection and classifications. So many scholars are working such type of designer drugs. Every year, new students come on choosing 2 or 3 drugs that is new and that is designer. That is the novelty is that that drugs is not reported previously. Okay. Such type of NPS. Okay. Now, global challenges in forensic toxicology, that is analytical limitations. Like when I work in Nepal, that was one of the developing country, there was thin layer chromatography for the purpose of preliminary examination. But when go for the confirmatory, they again go for the TLC. Why? There was not trend people. GCMS is there is not proper electricity, not trend people. Okay. So we require 2-step analysis, that is preliminary and confirmatory, in any legal cases to finding the proper results, and that is accurate results for the purpose of law. Regulatory hurdles, there is a proper regulatory hurdles required. Like in India, there are different toxicology societies, like Indian Society of Toxicology, talking mainly in the medical toxicology. There is STOX, that is talking mainly about industrial toxicology, environmental toxicology. So there are many associations, they are talking problems related to toxicological issues, and still such type of cases is in the notice of our scientists and professors. Resource constraint, limited access to cutting-edge forensic toxicology and expertise. Particularly a challenge is low resource regions. What happened when I chose MSc Forensic Science? I was very interested to pursue my PhD in DNA profiling. DNA profiling lab is very sophisticated, very clean. And in my admission prospectus, I read name of Dr. Anupama Rana, ma'am is here. So during my master's, I thought I will do my PhD under madam, but I never met with ma'am in 2006. So I also went for 6 month training in DNA profiling at CFSL Hyderabad, also cyber forensic. Then I got opportunity to teach in different medical college of Nepal, and I was 7 years there. So there was toxicology was one of the part in forensic medicine. And again, one of the director, Dr. S.K. Shukla, he was yesterday, there was, his lecture was there here. So he proposed me to do PhD. So I listened, but after 1 or 2 years gap, I registered my PhD with specialization in forensic toxicology. So still my students, my PhD scholar work in forensic DNA profiling as well as forensic chemistry and toxicology. So as a professor, I required to teach every subject many times if faculties are not available, specialized people. And but we have one major specialization in every field. When I presented one of my paper, yeah, during 2000, during 2013. So Goria sir is here. He also told always focus on one specialization. Okay. So in forensic medicine, what happened? People go for dissertation in their last year in one specialization, and in master in science, master in forensic science, we required many times required to read each and every subject. Yes or no? Like first time when first I joined College of Medical Sciences, Bharatpur, Nepal, and second, National Medical College, Birganj. So my principal told that you are teacher of forensic medicine, you're required to teach each and every topics. So many times our older professor not offer some subject, they just wanted that I will teach such topic, you will teach such topic. So this is my good things that I taught each and every topic in forensic medicine, and again joined Amity Institute of Forensic Science in 2015. So my thing is that specialization you required, put one specialization, but also you require idea about your other subjects. Okay. And as a director or a leading person, when you are giving some opinion, you require the subject, you require idea about that subject. If you are director of a one center forensic science laboratory, you require the knowledge of basics of each laboratory because you are director. And for the specialization, you required to know each and everything in that field, what happened in that particular field for your expertise person. Not for you, that you are expert, for the society, for the person who required judgment in the, they required justice. If you are not expert, you are not expertise in that field, so that is not bad for you, that also bad for the society. So always choose one specialization that is very necessary and work in that field. Okay. Then ethical concern. Ethical concern when we are studying about forensic medicine, jurisprudence. Okay, what happened? There is a doctor-doctor relationship, doctor-patient relationship, doctor-state relationship. So in forensics community, there is also proper respect required. Suppose one person is the forensic toxicologist, just finished his or her PhD 1 year back, and some person is 20 years of the experience. Okay. So what happened? That person had 20 years of knowledge, but that person who had 1 year of experience, that is also your colleague, and that is the in the field. So they also know very better about recent advancement than 20 years of experienced people. So proper respect and ethics is required. That proper ethics requires that you give the respect. And also if you are new and you know everything, is also required to give respect to your seniors. Okay. And never tell that my result is better than his results. You are not go inside the instrument and give the results. Yes or no? You examine with the help of the instrument. You are for the basic examination, TLC, then HPTLCR, GCMS, LCMS. What you are using, that is analyzed from the instrument. And thing is that if you are examining, yes, experience will be helpful in cases of interpretation. Your interpretation is very well than the new one maybe. So proper respect and ethics is required. Okay, like in doctor relationship, what happen if you are already treated by one doctor and you are not cured? So doctor is not going to say he is don't have proper MBBS degree or such type of things. Okay, okay, no issue. His experience, his or her knowledge is different, or different lab, different way, but proper treatment is required by another doctor without saying anything. But in forensic science, this is the bad thing is that I am the big scientist, you are the small scientist, such type of issues. So ethical issues is a very big things you require to give proper respect to each and every people. Then prevention and solution, public awareness campaign is required. Preventive and social forensics. So preventive and social forensic toxicology is also required. Just like preventive and social medicine, prevention is better than cure. So already some association like IST, they're talking about household poison. So if household poison, that household, so you just put such type of poisons from the not reach from the children, that is inside the some boxes and everywhere. I also mark that is the household poisons, like there are different infectants used in houses. Okay, so proper public awareness about exposure of pesticides. Okay, proper awareness about treatment of snake bites. Okay, uses of anti-venom. So such type of things required. And there is poison centers is also in different medical colleges giving the idea about awareness. Also, they have pamphlets, brochures for such type of awareness, and that is very needful. Okay, improved access to toxicology testing, like if particular regional forensic lab don't have that facility, then forwarded to the central forensic science lab to examine such type of things. Or generally what happen in forensic science laboratory, that if that is not in the scope of that lab, so that forwarded to the another sophisticated forensic lab. Regulation and control is required, and such type of things are going on and preparing in a better way. Training medical professionals. Actually, what happened, sometimes analytical part is very weak for the clinical professors, and sometimes there is a medical knowledge weak in the analytical scientists. So there is a proper training is required, both professionals. Then innovations in forensic toxicology. Okay, these techniques, there are different techniques like LC tandem mass, GC tandem mass, and there are limit of detection and limit of quantification. Okay, is required for each methodology, each instrumentations. Okay, then biosensors are very helpful. Like I already told, I have one copyright in date rape drugs and assaults related to influence of DRDs. So proper biosensors is, some nail polish is having that capacity. If that going to, okay, inside the that drink, that color is going to change. So persons know that is the such some adulteration or some mixture in that drink. So that lady, that girl will not take that drink. Okay. AI and machine learning. Many scientists, many researchers also work on the easily finding of poisons or drugs, that is the banned drugs, okay, on social media like Facebook and other places. So there is a sale of such type of products. So there is a proper prevention is required, and AI and machine learning can help not in as a wet lab, but for the data purpose. Okay. Omics technologies, toxicogenomics, and metabolomics. So what impact of that poisonous substance to at gene level, that is also a study is going on. Then lab on a chip devices. So like preliminary examination, before preliminary examination, we go for the spot test. Like in mobile forensic van, we can use such type of chip devices to know there is any poison substance or toxic substance in cases of narcotics and psychoactive substances. If that is in the white powders, so we can use such type of chip devices. Okay. Accreditation is required, like many forensic science laboratories that is not NABL accredited. So that is enhance the quality of that laboratory, that is international level. Okay. Standardized protocols is required, and also proper training is required. Okay. Ethical compliance, everything's in, like as an NABL assessor, I found NABL has all criteria to make a laboratory at global level. Okay. One case, one of my students working on some psychoactive substances, and that is spiking and stupefying. Like in jurisprudence of Modi book, also there is mixing of some psychoactive substances in tobacco or other drink or other food products. Okay, so stupefying is also, and also there is assault. So such type of cases happen. So she just developed extracted with the help of liquid extraction methods, and also developed one extraction methods. Okay. And also has spectra on that. She is going to submit her PhD in August 2026. Okay. Thank you. I want to acknowledge my mentors and all national, international quality toxicologists, professor, organizing team. This is some membership in the Forensic Toxicology Society. Okay. Thank you.
Thank you, sir, for that excellent and enriching presentation. Respected session chair, distinguished speakers, and dear participants, as we come to the close of this insightful morning session, I would like to express our sincere gratitude. We are deeply thankful to our esteemed speakers for sharing their valuable knowledge and enriching us with their expertise. I would also like to extend heartfelt thanks to our respected session chair, Dr. Anupma Raina ma'am, for her constant guidance throughout the flow of the session. A special note of appreciation goes to all the participants for your attentive presence and active engagement, which truly made this session meaningful and interactive. With this, we break for lunch. Please enjoy the meal and do join us again for the post-lunch sessions. Thank you. 150. Please be here at 1:50. Thank you.
बंद कर दो ना
शिवानी
I. Hello? Hello?
बहुत बढ़िया
Thank you so much. I need 1 minute. Is it okay? Can you see my presentation?
Yes, it's there.
Okay, thank you so much for invitation. I'm really glad to be here and to do my report. Actually, it's not about India climate conditions, but it is very important for our country to investigate gas-headed formation and some new ways how we can storage and transport gas in our cold region. So I think that's why it also might be useful for you to find, to clarify new scientific direction for investigation, especially if you are a young scientist, or for example, a student. So no, the topic it sounds like this, but we can touch many other things. As you know, Russia it's cold region in most territory, and so that's why for our oil and gas companies, it's difficult to explore oil and gas from frozen ground. So that's why we need to understand how we can do it in the best way. First of all, and then after exploration gas, we need to store it and transport, and that's why we need to investigate new ways how we can transfer and store it in permafrost regions, or for example, Arctic zone or cryolithic zone. It is all the same territory. Companies understand they can use several forms of gas for transportation and storage. First of all, it is gas in gas state, and we can use pipeline. But here we can compare some other ways. For example, liquefied natural gas, how to compress natural gas, and how we can transfer and store gas in solid state. And all these methods are used in our country, but solidified natural gas only as experimental technologies. It is not used in the company's objects, just in the lab. And liquefied, it is good method, but it's too expensive. Compressed, it might be usable only for several less quantity of objects. And so that's why companies are more interested to produce their own ways how to storage and transport gas, for example, in solid form. There are some problems, some issues where a company can realize and use this new method. For example, first of all, it helps us to understand how to explore gas from gas hydride if we will investigate gas hydride as an object in the lab. The second is to develop new technologies, how to utilize or sequestrate CO2 or petroleum gas. No, one more picture, it is about CO2 sequestration. And now we are thinking about the development and build temporary storage of natural gas in solid hydride state in permafrost region. We have some this kind of storage, but not for gas, but for seeds. It is also important for settlements and for it's like state task how to store seeds. For example, if we are going to use them in 100 years, or for example 200, several hundred years. If you are not in gas hydride topic at all, I can tell you that what is it gas hydride? Methane hydride, actually. Methane hydride can form from molecules of gas and water at special thermodynamics conditions. For example, at high temperature and low pressure. That's why in the regions with low temperatures, these regions with low temperatures looks like good places for gas hydride formation, because we can save money and other resources to transfer gas to solid state. But in spite of for gas hydride formation, we need only gas and water. It is very difficult to form gas hydride from gas and water. And it is one of big problems why so many scientists, not only in Russia, investigate gas hydride, how to promote gas hydride formation, to synthesize some new additives that helps us to increase gas hydride formation rate. And the problems, no, we have several problems. Low rate of water to hydride conversion, low degree of water to hydride conversion. And I can tell about Russia that we don't have ready-made technological scheme for the transfer of gas to a solid hydride state for transportation and storage. We don't have ready-made technologies for companies. So that's why we think that we should receive some tasks. And for example, if we will not receive the first task here, we cannot start to receive the second. And the first is achieving the maximum rate of water to hydride conversion in the shortest possible time under laboratory conditions on the first step. Then we can try to scale it. To develop a mobile autonomous complex for converting gas to a solid hydride state, it is economically will be good for companies to use this kind of techniques, mobile autonomous complex, because we can use it in one object. In several years, we can transfer it and use in another object. To determine the methodology and technology of regasification, to understand all steps of this technology. Develop a step-by-step technology scaling plan from laboratory to industrial scale. And how this problem is solving now all over the world in different labs in US, in India, in China, in Malaysia, in Japan, and in Russia also. Scientists test and synthesize and try to check how these promoters works in the lab. No, first of all, promoters, different kind of promoters, thermodynamics, kinetics. One more way is how to increase gas hydride formation rate, use different configurations of high pressure vessels. And one more way is to produce dispersed system, and there also might be with promoters, there might be with some other additives, for example, additives which can stabilize this system. And this kind of systems, dispersed systems, can use multiple times. And this kind of researchers we observe here in Russia, and also there are some groups who investigate this kind of systems in China. And I know that in India also systems looks like dry water also was tested. What is our goal? And even it doesn't matter, we are talking about additives, we are talking about some kind of dispersed system. The main goal is to demonstrate for oil and gas companies the current state of the development of gas hydride technologies and the availability of new effective solutions that we have, not only in Russia, that there are all over the world these days. And now our tasks are, no, it is for this year and for previous and maybe for the next year, no, for 3 years, for example, To determine the effect of the concentration range of soy lecithin or other lecithins on the effectiveness of stimulating the formation of methane hydride formation from liquid solutions. Compare these lecithins additives with other well-known promoters. Now, for example, well-known promoter is the diethyl sulfate natrium and also polyvinyl alcohol, but only from frozen powdered solutions. Observe the influence of lecithins on the experimental sample characteristics and behavior temperatures close to ice melting, because at these temperatures near close to 0 degrees, we can observe the highest water conversion rate. And for this investigation, we can use nuclear magnetic resonance spectroscopy and also DSC, difference scanning calorimetry. And one more, it is investigate of gas hydride formation in powder cryogels. And the last one here, it is based on experimental data, compare different powder systems. No, looks, for example, dry water, powder cryogels, powdered gel, dry gel, sorry, for the synthesis of CO2 also, not only methane hydride, but also CO2 hydride. These are methods and equipments we use in our investigation and everyday work, and I told about them previously. It is nuclear magnetic resonance spectroscopy, PVT method, and we have different kind of high pressure vessels reactor with window, without window, with mixture devices, and so on, and different scanning calorimetry to investigate phase transfer. This also about methods, and this we can see how our experimental equipment looks like. Every experimental equipment has high pressure reactor with no, sometimes with, sometimes without stirrer. Sample we have in high pressure reactor vessel, temperature sensors, several in gas phase and in the sample, programmable cryostat, some other equipment. It's common for all experimental setups for investigating gas hydride formation, and nuclear magnetic resonance spectroscopy method. Here we can determine the quantity of water in the samples. And for example, if we have the samples that contain gas hydride, it means some part of water in hydride form. So other water we can see, and we can determine how much water transfer to gas hydride form. And we can do it at different temperatures. This is also about experimental setup where we can investigate CO2 hydride formation, and not only the process of formation, but also dissociation and self-preservation. We needed to understand and estimate how we can store CO2 in gas hydride form. It's most of all ecological topic about CO2 sequestration in hydride form. And this about materials, and soy lecithin, it is natural surfactant. It's made from no, it might be made of, it might be animal product, it might be plant product, and we are going to taste all of them. And it is absolutely ecologically friendly. So that's why it's better to use, for example, soy lecithin instead of diethyl sulfate natrium. And in our work is one of goal, one of task to demonstrate what is better to use, soy lecithin, for example, or diethyl sulfate natrium, or polyvinyl alcohol, which is better for companies to use, especially if we are going to talk about industrial scale. Here you can see the list of systems we tested and their components they we have inside. And for example, the simplest system is powdered ice. The second is dry water, dry gel, powdered gel solutions. PPVACG, it is polyvinyl alcohol clear gel. It is also powdered system. Solicitine solution, frozen solicitine solution, and powdered hydrogel system. And here you can see the view some of these systems. Most of them looks like powder, but sometimes it looks like some other substance. And the first step of investigation, it not only about solicitine solution, it common for other systems also. We need to estimate the stability system at constant conditions, at constant temperature, for example, room temperature, and also how the system behave before and after freezing thawing cycles. And here we can see the solicitine solution. First row, it is freshly prepared solicitine solution, and the second row, it's in 1 week after the preparing. And we see that the solicitine solution is stable during 1 week, and also we can tell that during the 2 weeks it is also stable. So it's good characteristics for this kind of additives, so it might be usable in companies. One more important thing for surfactant, solicitine is surfactant and SDS is surfactant, and we need to compare them, which is better for companies. No, for scientists it's both okay. We have a good rate of gas hydrate formation, it is okay. But we are working on companies and we should tell them what is better for them. SDS, it's toxic, solicitine is ecologically friendly, solicitine is stable in time, as the SDS is also stable, but there is one more factor what we need to compare, foam formation. We know that SDS, to form huge quantity of foam, it's the upper picture, right? It is right from the right side. It is SDS, and the left side is solicitine, and we see that solicitine doesn't form foam. So it means that solicitine is in this aspect is also better than SDS. And here we need to compare according to our experimental results, which is better according to the degree of water to hydrate conversion. And induction period of time, it is also important characteristics. We can tell that induction period the same for solicitine and for SDS, so we can compare degree of water to hydrate conversion. And we try to estimate water to hydrate conversion, the rate of water to hydrate conversion, the first 30 minutes of experiments and 400 minutes. And if we will talk about first 30 minutes, SDS is much better. So it means that gas hydrate grows from SDS solutions from the first minutes rapidly. But if we will talk about some other period, for example, 400 minutes, solicitine with the concentration 0.5 weight percent is also good. No, results are the same for solicitine and for SDS. So it means that after a long time, the systems behave the same. But on the first 30 minutes, SDS is much better. And here there is one more characteristics that we can calculate on the experimental data, gas consumption. No, here we also this results is the same because experimental data is the same. So we can see that solicitin solution with concentration of 0.5% E and SDS with concentration of 0.1% the same. And using a high pressure vessel with window, we investigate, we observe that gas hydrate formation rate, or the gas hydrate formation process from soy lecithin looks like in gas hydrate formation from SDS solution. Sorry. And this mechanisms has a special name, capillary-driven mechanisms. And first of all, gas hydrate grows on the high pressure vessel wall and then in the volume. What is also important, we try to observe gas hydrate formation from soy lecithin at different temperature. Because we have some idea that we will form gas hydrate in the company's object, and it might be at different temperatures. And most part of the year they have negative temperature, and we research water to hydrogen conversion ratio at different temperatures. And at -1 Celsius degree, the results was much better than at temperatures -3 and -5. No, it is just experimental results. We don't know at which temperatures companies will work, but anyway, we have this data, and then we can plan some other experiments. It is also interesting that some additives can increase gas hydrate formation only if we form gas hydrate from liquid solution. Some other additives, for example, polyvinyl alcohol, works only if we form gas hydrate from frozen powdered solutions. So it means we have solution, frozen it, powdered it, and then started to form from this ice powder gas hydrate. But soy lecithin from both kind of system, from liquid solution and from frozen powdered solution, demonstrate good results. So in our days, all over the world, we have only one this kind of system and kind of additives that can help us to form gas hydrate with high rate from frozen powdered system and from liquid solution. It's soy lecithin. This is about our nuclear magnetic spectroscopy measurements. We try to understand why soy lecithin can help us to increase gas hydrate formation rate. And for example, polyvinyl alcohol also. And we prepared several samples. All of those samples looks like frozen powdered systems. Frozen polyvinyl prepared from frozen polyvinyl alcohol solutions and prepared from frozen soy lecithin solutions. And also powdered ice. It's look like clear system. So we had three kind of systems, and we understand that the worst results was for ice, and ice had the lowest rate of melting, and polyvinyl alcohol has the highest rate of melting. And soy lecithin, it will be in the middle between ice and polyvinyl alcohol. And according to our gas hydrate results, it's have the same correlation. Polyvinyl alcohol give us the highest gas height formation rate. And now we understand that it is because of fast melting. But ice gave us the worst result, and soy lecithin in the middle. It is about powdered systems. We have some information and some idea according to our experimental results with powdered create gel and according to our results for gas height formation from polyvinyl alcohol and powdered polyvinyl alcohol with different concentrations on polyvinyl alcohol and stabilized additives. As stabilized additive, we use hydrophobic silica nanoparticles that were produced algal chemicals. And we find the system which gave us the best results. What does it mean, the best results? It means that we can form gas height with the highest rate, first of all. And the second, it means that we can use this system multiple times. For example, this system, we tested 9 cycles. And these results were compared with results that were observed some group of scientists in China. It is also about these systems, and we can see that it looks like one solid piece. Gas height looks like one very solid piece after formation, but on the first, no, before the experiment, it looks like powder. It is also about soy lecithin, but if we add some salt in water, so it means we solving soy lecithin in salt solution. Why we should do it? Because companies work with some kind of mineralized water, and we want to suggest them to use mineralized water for gas height formation, and we need to understand how it will work with promoters. We also investigate the stability of this system in time. And here we have some problems. Not all systems are stable. For example, if we will talk about two additives, soy lecithin and salt. And of course, these are our experimental results, kinetics, water to height conversion ratio, and we can see that we can compare just distilled water, soy lecithin solution, and the soy lecithin solution, how it works with mineralized water. And we can see that soy lecithin solutions, as we see previously, started to give us the higher water to height conversion ratio at the end of experiment. But if we add some salt, we have the highest rate of gas height formation from the first minutes. So we can tell that if companies will use mineralized water, it will give them much better results. But we need to compare it several times, and we need to vary some other samples of mineralized water from different companies' objects to clarify these results. And this about CO2 sequestration, how to form CO2 gas hydrate, from which systems it would be better to form CO2 hydrates. And we tested several kinds of systems, and here we vary stabilizer. Stabilizer, it means hydrophobic silica nanoparticles with different properties, and we use R202 and some other. And also we add some promoter, for example here soy lecithin and SDS, to this system to check which result might be better. And we can see that row 7 and 8 gave us the best results. This looks of these systems. For this system, we understand that it works better if it is powder. If it is not powder, it will not work better. And here we see also again experimental results, and we can see that water have the worst result, and soy lecithin for it is for CO2 high deformation. And other systems, dry water with additives, gave us the best results. And no, here also about these systems, and also with calculating amount of stored gas, if it will after investigation of self-preservation, when we drop pressure and storage this system and this at these conditions, and then we can estimate how much gas store in gas height. And that's all. And I want to tell you about the conference that will be in Russia, in nice place Kaliningrad in September, and we'll be glad to see you or your colleagues on this conference. Thank you so much.
Thank you so much, ma'am. If you guys having any question, you can ask. If वो करेंगे। Okay, meanwhile, sir, I will ask question.
So the kinetic data, did you fit it to any model, or like could you get the rate constants out from the kinetic data?
It's a good idea, and we have in our lab person who do some modeling, but it's not about these experiments. It's just some other modeling. Here we don't have enough experimental material for modeling because they are very different in different conditions, and we just do some search the best additive. And then, well, when we will find the best additive, we can model.
Okay, thank you.
Thank you so much, ma'am. It was a very informative session, and we are very humbled and privileged that you joined us in our international conference, and our audiences has enjoyed your session. Thank you so much for joining.
Thank you so much.
Thank you, ma'am. So here we are. I am deeply honored to welcome our guest of honor, Dr. Pratishta Pandey, Head R&D Infrastructure and NSM. Ma'am, are you there? Are you able to listen us? I would.
Hello, good afternoon to all of you.
Yes, ma'am, you're audible. And I would like to invite our director, Amity School of Applied Sciences, Professor Seema Pathak, ma'am, to welcome you and introduce you.
Please clap little louder. Ma'am is there. So, ma'am, hello, ma'am. How are you? So
I'm good. Yeah.
we are deeply honored, ma'am, and we are welcoming to you, Dr. Pratishtha Pandey, Head R&D Infrastructure and NSM. Dr. Pandey is associated with Department of Science and Technology, Government of India, where she serves in area of R&D infrastructure. Her work is connected with the strengthening research ecosystem, scientific instruments, and national mission driven programs, including initiatives relevant to advanced research and technological development. It is our privilege to invite Dr. Pratishtha Pandey to share her visionary remarks with us. Ma'am, may I request you to kindly address the gathering?
Dear faculty from Amity University, from nearby academic institutions, researchers, students, good afternoon to all of you. First of all, I would like to thank Professor Seema Pathak, who gave me the chance to address this distinguished gathering at this very important international conference on frontier areas of science and technology for sustainable development, which is being organized at Amity, Gurgaon. I think this platform must have provided a very good opportunity, especially to the young researchers, to discuss new ideas in science and technology and their research fields with eminent personalities who have come and delivered the talk at this conference. So, first off, I would like to brief you about the progress which Indian science and technology has made. Modern India has a focus on science and technology. So, we are the third largest education ecosystem in the entire world. And it is very encouraging to note that Indian science and technology has progressed a lot. It has made huge progress over the years. Now, India stands third in the number of publications, in the number of scientific publications, third in the number of PhD produced. India is standing third in the number of startup ecosystem, and also the woman participation has doubled over the years. Today many important directors in leading institutions in different CSI labs, DRDO labs, many secretaries in government of India, and various research groups, leading research groups in the country in different academic institutions are led by women. So these are very important trends in the Indian science and technology. India has a very large, huge workforce in STEM areas, and also India stands third position in the number of space exploration. And also, I would like to mention that the Global Innovation Index has also jumped over the years. Now it is 38 in 2025, which was earlier 81 in 2015. So these are very important and very progressive trends in Indian science and technology. And we all know that one of the mandate of government is to build a very strong infrastructure in academic institutions. So today, science and technology are the heart of sustainable development. The global challenges we face on climate, energy security, and public health require integrated solutions supported by strong research ecosystem and advanced infrastructure. So, Department of Science and Technology, as many of you must be having DST grants and sponsored grants, is providing research infrastructure in different academic institutions. So you must have heard about FIST. You go to any corner of the country, you will see a footprint of FIST in different academic institutions, in various IITs, ISERs, NITs, then private universities, state university, and also in central universities. FIST has been provided in different campuses of Amity also. I think Noida campus, your Manesar campus, Amity Gurgaon, they have got FIST funding over the years for in different departments for strengthening the research infrastructure. Also, one very important program which was initiated by DST in 2009 is the PERS program, where huge quantum of funds are provided to the university ecosystem for strengthening the research. And till date, 91 universities in the country, they have got PERS grant. These are the performing universities in the country, and in different thematic areas, the grants are provided. Also, I would like to mention the other program which is I'm handling is the National Supercomputing Mission. So in National Supercomputing Mission, DST along with MIghty, 39 supercomputers are installed in the country. You can visit NSM India website to know the locations of the supercomputers which are there in different academic institutions, and they can be accessed remotely also. You can visit the NSM India website, create an account of yours, and those, I think many of you are coming from theory background, you should know what is the where the country is going, where are the supercomputers in the country, and how do you access, how do you avail, strengthen your research by acquiring these facilities for your computational requirement. I think this conference organized by Professor Seema Pathak of Amity University must, I think the young, especially for the young people, students and researchers, you must have got very good insight from various talks from various eminent academicians, and also many international speakers also presented their talk on their research topics. And I think all of you must have also got chance to build to learn from them what are the latest trends in science and technology and your research fields. And with this, I will not take much time, and I wish all of you a very good future for your research and endeavors. Thank you so much.
Thank you, ma'am. And I must appraise you, ma'am, that while during this conference, we have 40 speakers. We were having 4 different panels for chemical, physical sciences, mathematical sciences, and forensic sciences. And we were having 40 speakers. Of course, some of them were from 13 speakers were from foreign international universities, and rest of were from Indian. And one of them, yesterday you just connected, Dr. D.S. Rawat was our chief guest. And in that way, we have got blessing from every corner of the scientist, and from you also. And definitely this will give us lots of motivation to perform further and to appraise you that we have got, as you mentioned, that Amity University has got many fists, but we are lucky one. We have 3 fists, right? We have got 3 fists, and I think let's clap for Amity University, Haryana, and for the support rendered by Department of Science and Technology for granting and believing and having faith in us. And we have developed infrastructural world-class labs. I am assured that definitely any day you are there to visit us and to see that how much hard work we are putting in and how much we are helped by the management of Amity University Haryana, our founder president sir, or this Chancellor sir Dr. Asim Chauhan, our Vice Chancellor sir Professor P.B. Sharma, and our Pro Vice Chancellor sir Professor Dr. Vikas Madhukar, and Dean Science Technology Dr. Rajendra Prasad sir. So we are blessed, and we are eagerly waiting for you to come to next time to Amity University Haryana. I hope that we are going to meet soon. Thank you so much for devoting your time and inspiring our students in such a deep way. Definitely they are going to rock the world in science and technology. Thank you so much, ma'am.
Thank you so much. Thank you.
Now I request Dr. Priyanka to continue session further.
Thank you so much, ma'am. We shall now proceed to our plenary session 4 by Professor Ankush Vij from Central University of Haryana. It's my privilege to introduce Professor Ankush. He is a professor and head of the Department of Physics and Astrophysics at Central University of Haryana, Mahendragarh. He has held academic positions at UPES Dehradun, NIT Hamirpur, Amity University Haryana, and the Korea Institute of Science Technology. His research focuses on nanomaterials for solid-state lighting, energy harvesting, sensors, and radiation dosimetry. He continues to advance sustainable nanotechnology, and serves on the Academic Council and University Court. He'll be delivering his session on optimization of luminescent materials towards sustainable lighting solutions. May I request Professor Ankush Vij sir to deliver his plenary address.
We have to share this in Zoom as well.
Sort the slides from here.
Yes. We do have a mic, but since the conference is on the hybrid mode and you can use the podium mic so that the online participants can also listen. Yeah, if you will be roving, then they won't be able to listen.
So
Ma'am,
thank you.
slides, slides are not in a presentation mode, on online mode.
We have already.
It's not in a presentation mode on online. There you must be seen, but right now it's on online. You can't see.
So a very good afternoon to one and all present here. So first of all, I thank organizers, especially Professor Seema Pathak Madam, Director, MIT School of Applied Sciences, for inviting me at AUH. And to begin with, I just would like to recall my memories connected with AUH. I started my career at AUH in 2010 to 11, and in 2015 to 20, I served here again. So all in all, 6 years I have spent in this beautiful place. So it's really refreshing and very good to come again and meet old friends here, and also presenting some of the work that which I am carrying out in our lab. So to begin with, I'll be briefly discussing on luminescent materials for different applications. So currently, I'm also heading Department of Physics and Astrophysics at Central University of Haryana. So this is our university. The upper one is the administrative block, and the lower one is the block one, which is science block, and the Department of Physics and Astrophysics is located in this building. And just to begin with the glimpse that what we are doing in our department. So at present in our department, we are offering 3 courses, including a PhD doctoral program. And then we have Masters of Science in Physics program. And then we have one integrated BSc MSc Physics program. It is 3 + 2 years. And now as per NEP, it is changed into 4-year undergraduate program. And the major research facilities that we have in our department, we have one UV visible NIR spectrophotometer, which ranges from 200 to 3200 nanometer. We have DTA TGA, we have gas chromatography, thermal CVD, microwave etching furnace, high temperature furnaces, combustion furnaces, tubular furnaces with environments like argon, nitrogen, argon plus hydrogen. We have recently developed one gas sensing setup also. Source meter we are having, and these red ones which you are seeing here, I don't know, it's not okay. So we have these 3 facilities: RF magnetron sputtering, temperature dependent micro Raman, and photoluminescence setup with up conversion and phosphorescence decay, which we received as a grant from DST as a FIST program, and in which the magnetron sputtering and photoluminescence system is already been ordered, and micro Raman is likely to be ordered within next 1 month. So these 3 facilities we will be having very soon, maybe next 3 to 4 months. And apart from, so it's not moving. Double click करो. So apart from this, We are also in the process of having central instrumentation center, whereby Ministry of Education have granted around 100 crore for this project, and soon we will be having many facilities, some of which are in ordering stage and some of which we will start in the next phase in the next 3-4 months. So the major facilities that we will be having in this center is HRTEM, FESEM, PPMS, XPS, probe station. These red ones, they're already in the purchase process. And the other ones, their purchase will be started soon. So all in all, these systems will be installed in CIC, which will not cater only our university, but the neighboring university and within North India also. So this is the mandate of this Central Instrumentation Center. That's why I wanted to show it to you. So now coming to the topic. So I'll be starting briefly with the basics of luminescence and phosphors, and then introduction to some sustainable lighting solutions. And finally, why we need to go to search these materials, what are its applications, and then some experimental results and discussion. And then finally, I'll be concluding with what recent developments have been doing in this field across the globe. And then finally, I will be summarizing. So quickly, if we see that how does light interact with material optically. So we have various fundamental processes going in here, like we can see this I naught. I naught is the, if we consider this I naught as the incident light, then how it can interact with the material. It can either transmit through the material, or it can get absorbed in the material, or it can get scattered from the material, or it can get re-emitted in the material, from the material, right? So all these processes, they constitute some different experiments. So majorly in our research area, we are more concerned about this IE part. So IE part is the emission part. So emission part means when this emission is in the form of some radiation which spans in the visible region, this is what is called luminescence. So luminescence essentially, you have a material, you excite it, and then upon de-excitation, if it gives the visible light, it is called luminescence. And based on several factors, we have different kinds of luminescence. For example, we have photoluminescence, we have chemiluminescence, trivo, bio. So it's a basically different modes of energies that you can give to a system, and in return, you get the emission back. So I will not go into detail of this, just a glimpse to start with off. So why these materials are important? Because these materials have lots of application ranging from our day-to-day life to the state-of-art machines. For example, you might have heard of fluorescent bulbs, right? And then we have solid-state lighting. And then we have long afterglow storage phosphors. We have radiation dosimetry. We have medical imaging. We have paint, signages, up conversion, cathode ray tubes. So n number of applications we can have from this phenomenon. So primarily, I'll be talking about a solid state lighting, which is also called LED technology. So if we see the chronology in lighting solutions, so we come to know that in 1879, we had this Edison light bulb, and now up to 2010, this has changed to the white light emitting diodes. So LED technology has taken over these earlier technologies. So what was the problem with the fluorescent lamps? The fluorescent lamps, basically, they are the mercury-based discharge lamps. Then their emission line is at 254 nanometer. So 254 means they emit in the UV light. And these lamps, they are coated with a layer which absorbs this UV and then emit back in the visible region. But their efficiency is limited by this equation. So we can never improve, or we cannot improve its efficiency over 25%. Right. So therefore, there was the quest of developing some other technologies in this field. So therefore, thus it is not considered as a sustainable lighting solution. So for a sustainable lighting solution, they should have minimal environmental impact while maximizing efficiency. So therefore, the key components, which includes, they should be energy efficient, means to reduce the amount of electricity needed for illumination. They should be environmental friendly. They should last longer, and they should have recyclability and disposal. So these are some of the properties which any sustainable technology should have. So is the lighting solutions should have these components must be included. So therefore, the role of LED technology becomes really important to address these problems, because how these light emitting diodes improve energy efficiency in homes. So we can see LED uses up to 80% less power. So they are energy efficient. They have long life. You could see the LEDs have a general lifespan of 25,000 hours or even more. So it is much, much greater than incandescent sources or the CFL sources. And also, they have flexibility of varying the color temperature, means dimmability you can play with. So all these ingredients are there for sustainable solutions for this LED technology. So therefore, this solid state lighting, which is a topic of the day, becomes really very important. So this SSL refers to a type of lighting that uses LED as a source of illumination than electrical filament to produce white light. And what is the advantage of this? Because it is highly energy efficient, it's a long life, and also it's cost effective. Right. So ultimately, if you see this CIE diagram, so we can correlate the color temperature of any LED produced by using this CIE diagram. So generally, those who work in the white light emission materials, their output must lie somewhere here in the white light or the red emitting LED or the green emitting LED or a blue emitting LED, depending upon the different applications. So the problem with white LED is that generally blue LEDs are used and some blue light escapes. For example, here I have shown in the simple carton, you can see here, this one is the blue emitting material. This is a blue LED, right? This blue LED is coated with some layer of a material which emits in the yellow region after absorbing blue. So blue is changed to yellow, and blue plus yellow gives you white color. But the problem with this white color is that if you compare it with the sunlight spectrum, so this is the perfect sunlight spectrum, which peaks at around blue region. But when you mix this blue and yellow, right? So this, you can see this 485 nanometer around this peak, this portion is missing from the white light. So this is deviated, much deviated from the diffuse sunlight spectrum. So therefore, there is a need to change this technology. So later on, 3 different color LEDs, their output were mixed. For example, scientists have taken blue LED, green LED, and red LED, and they mixed them to get the white light, which is close to the diffuse sunlight spectrum. But again, the problem with this technology is that to deal with 3 LEDs and their output is very difficult to mix and to have a control over them. And also, these 3 different LEDs have different degradation time. So over a period of time, again, the chromaticity of white light, it gets destroyed. So then there is another approach which was adopted by scientists, that why we can't have a single material which when coated over a blue LED, it emits in the white light. So therefore, the goal ultimately becomes develop single phase suitable direct white phosphors. So this is the essence of doing this kind of research. So once you know which material to be tested, then there comes the testing part, the experimental part. So I will be just touching upon one experimental aspect, which is photoluminescence aspect, because we keep on testing these materials using photoluminescence spectroscopy. So for example, in PL setup, we have 2 kinds of spectra we can have. PL emission spectra we can have, and PL excitation spectra we can have. So this is a simple experimental setup where we have a light source. We have one excitation monochromator. This light source emits multiple wavelengths, and using this excitation monochromator, we can select wavelength one by, we can select this lambda excitation, which is made to fall on this sample. And once the sample gets excited, means sample gets its suitable wavelength where it can be excited. So again, it has to emit back. And if that emission is recorded again using this emission monochromator on detection side, then it is detected using a suitable detector, and you get a peak between the intensity versus wavelength. So this is your PL emission. Conversely, if we fix the PL emission in the detector side and then vary all these wavelengths, we can have one PL excitation spectra. So this PL excitation spectra is different than absorption spectra, in a sense that here the excitation wavelength means that at every absorption wavelength, you will have an emission. However, the absorption bands in UV visible spectra does not necessarily lead to the emission spectra. So this is the one difference. And the most important part here is to choose the light source very wisely. So I have here plotted the the output profiles of three mostly used xenon lamp, mercury lamp, or D2 lamp. So why it is important? Because many a times these sources, the lines from these sources, they get reflected and appear in the emission spectrum, and which are ultimately taken as the data from the sample, but it is not. This data is from the source. So this needs to be eliminated. So this data analysis should be done very carefully. So this is PL micro Raman system based on laser. So this source, which I just discussed before, it can be a laser or it can be a broad source also. So both separate, they are separate experiments. Laser-based PL has its own advantages, and broad source-based PL has its own disadvantages and advantages also. The major disadvantage is that the flux is very high in case of laser, but with laser-based PL, you cannot have a PL excitation spectroscopy. But with broad band sources, you can have both PL emission as well as excitation spectroscopy. So this is a broad source based system, and this is laser based system. So as I discussed you that we will be having soon one advanced PL lab at CUH. So we will be having two systems for these PLs. One is steady state PL spectrophlorometer, which will be a departmental facility with a signal to noise ratio 10,000:1, which is generally 5 times higher than any normal machine, and which will be equipped with a 150-watt xenon lamp, and we will be able to monitor both PL emission as well as excitation. And we will also be able to measure lifetime measurements from microsecond to few seconds. So it means we can study all the lanthanide or actinide materials using this microsecond to few second lifetime measurements. And this system will have phosphorescence scan also. And more importantly, because if you want to take your research to the commercial level, you need to measure the absolute quantum efficiency. So with this system, we will also have this PL quantum yield measurement system. And additionally, the one advanced feature will be that we will be able to monitor this photoluminescence using low energy NIR laser also. So which is called generally upconversion. And the other system which we will be having at CIC, it will be even having better this resolution 20,000:1, and it will be equipped with picosecond time resolution. So it will be a time resolved fluorescence also. So in order to investigate these systems, such systems, the balance state is very, very important. So therefore, we make use of synchrotron-based X-ray absorption spectroscopy, where we use mainly this Zane spectroscopy, which is X-ray absorption near edge spectroscopy. And using this, we discuss about coordination chemistry, and we also calculate balance state, which is very important in such systems. So I will not go into details of this. So now just coming to some of the work that we have done. So I will not go into these XRD systems. Straight away, I will come to the results. We have developed a strontium sulfide based phosphors or based luminescent nanomaterials in our lab. Using strontium sulfate, we have reduced them to have a single phase strontium sulfide. And we performed their X-ray absorption spectroscopy in South Korea, in Pohang Accelerator Lab, in order to investigate their local electronic structure. And what we found, that you can see the first graph, and the system is a samarium-cerium co-doped system. We have doped both cerium and samarium in strontium sulfide, and we have seen that using this spectroscopy, that the balance state of samarium is 3+, as well as we investigated the balance state of cerium, which is also 3+. So, and simultaneously, we also measured DRS, diffuse reflectance spectroscopy, PL excitation, and PL emission. So you can see here the strontium sulfide cerium, it emits in blue to green region. So, So these two regions have been assigned to cerium levels. These are transitions 5D to 4F transitions. The 5D breaks into EG and T2G bands, while this 4F splits due to spin orbit interaction. And then we have these two transitions possible only. Other transitions, they are forbidden. Now, when we checked the photoluminescence of cerium and samarium co-doped PL, we found some interesting results. So these results are that a single system upon 3 different excitation emits 3 different wavelengths. So you can see here, when the same system, it was excited using 430 red one. So it gives you a peak like this, so which emits in this blue to green region. When we excited the same system using 335, this is blue one. So you can see here the profile totally changes. Now the emission is the sharp emission, and this broad emission on the lower region, it gets disappeared. So it means the emission is only due to samarium emission. So there is a switching of color from blue-green to the red part. So when we also investigated using PL excitation spectroscopy that there is another band which is at 393. So you can see here this 393 band. So when we investigated the PL emission at this 393 band, surprisingly, this is the black curve you can see where both the features lies. The feature of this red one and the feature of this blue one also lies here, and its output goes more close to white light. So we were able to deduce this excitation band and investigate experimentally that this blue light can be switched to a white light by just simply optimizing different parameters in these materials. Further, we tried to increase their quantum yield by various substitutions. So in strontium sulfide systems, we partially substituted strontium with calcium and barium, and we were able to increase the efficiency by almost 2.5 times. So in this way, we can see that there are different optimization ways whereby we can increase the switching behavior as well as the quantum yield of these systems. So now these are some latest results of one of my PhD scholar. I will not go into details of them. The major finding was that with the system of magnesium oxide Y2O3 composite system, which was co-doped with erbium ytterbium and holmium ytterbium, we were able to make an invisible ink derived from nanoparticles, these nanoparticles. So invisible ink means then how can we read it? We can read this ink, whatever we write with this using this ink, we can decode it using NIR rays. So whatever you write with this ink, you can only read through when you have NIR camera or under NIR light. So such ink is used for security purposes in defense sectors. So you can see here, so different words, because that student was registered in my earlier institute UPS, so she had worked over there on this problem. So similarly, we were able to have white light based on this up conversion also. So when I say up conversion means what? You have a system, you are providing low energy to the system, but in return you are getting high energy, means you are exciting the system with near infrared, but you are getting the visible region. So we were able to tune again white color using this up conversion process. And this you can see that the output is nearly this here in this white region. And there a switching has been found again from blue to white. The one output is in the 280 nanometer. It's a blue region. and just a switching, it switches to this 96 around. The output is in the white region, and you can see this paper, this is the latest paper of converted white light emission in erbium doped MgAl2O4 nanocrystals. So this is one of the work, and in other work which we published in Journal of Materials Chemistry C, here again we have shown that excitation energy dependent switchable emission in SRZNO2 system nanophosphors, and we have done some of the experiments in South Korea for this system, and some of the experiments they were done at UPS Dehradun with upconversion machine. So you can see here again blue to white switching was evident in this system also. The output in the A, you can see it lies in the blue, and output in the C, you can see it is near perfectly to the white CIE diagram. Similarly, we have done checked such properties on another system, which is inverse spinel, zinc, magnesium, aluminate system, and whereby we have modulated color by selective excitation activated, which was published in Dalton Transactions. And very recently, just 2-3 months back, we have been able to publish in nanoscale the same upconversion material. So nowadays we are focusing more on upconversion and its applications. So I would like to thank my collaborators, especially from Korea, where I have worked as a postdoc also and as a visiting scientist also. And also I would like to thank our Vice Chancellor, Professor Tankeshwar Kumar, for providing us the various opportunities to perform experiments and also to showcase our results in terms of allowing us to go to various conferences. And these are some funded research projects that I have been working in. And finally, I thank my doctoral research students. The red one, all they are doctorate, and these black ones, they are currently pursuing. The last one, Miss Sakshi, a thesis is submitted. So finally, thank you. Thank you very much. So if you have some queries, I would like to answer them.
Thank you, Ankush sir, for such an informative talk. If you guys have any question, you guys can ask. Yeah. Sir. Anirban sir, mic.
Although I know I was a bit fast because of 20 minutes time, I expected a little more.
But you have shown a very good.
Good evening, sir. Sir, आपने एक चीज explain करी थी that there is a difference between the absorption spectra as well as the excitation spectra. Can you please repeat it again?
Sure, sure.
Yes, sir.
Can I? So, okay. I think it's not working. See, excitation spectra, we record in PL emission experimental setup. The absorption spectra that we record in the UV visible spectroscopy. In principle, both are same. But while recording experimentally, both are different. Both are recorded in different machines. While we do UV visible, we get every absorption band in UV visible spectroscopy, right? But when we do excitation spectroscopy, only those absorption band appears for which there is always an emission. However, conversely, it's not true. Whenever you have multiple bands in UV visible spectroscopy, all absorption does not necessarily lead to emission. There may be some non-radiative decays. There may be some phononic decay modes, but every absorption does not lead to emission. Conversely, every excitation leads to emission because you are recording excitation when you are fixing some emission wavelengths in the detection side.
Okay, thank you, sir. And secondly, sir, which software do you use for the CIE plotting?
There are multiple software. They are only freely available nowadays because the one I used 10 years back, they were something different. Now the scholars they are using, they are something different. But they are freely available.
Okay, okay. Thank you, sir. Thank you so much.
In which material?
So do you do lifetime studies also for these
No, lifetime I have not shown yet.
Okay, so but do you know that
Yeah,
it's
it changes. It changes. We have noticed it using synchrotron-based studies because what happens? Certain states near the surface they change from 2+ to 3+.
okay?
But they do not show their signature in luminescence, which is more little in-depth study. So they have very negligible contribution in terms of luminescence. But obviously, balance state changes at the surface.
So maybe if you do XPS on the versus XRD, but bulk versus surface, that behavior
No,
may be different.
With XCS, we have found this.
Okay.
X-ray absorption spectroscopy.
Sir, I also have one question.
Yes.
Sir, you have choose with SRS2. Is there any reason of
No,
choosing
SRS and MGY203.
Hmm.
So there are multiple systems where we are working on with different scholars. SR is
Means like that SR choosing the strontium.
strontium sulfide.
Strontium sulfide. So that I just want to ask why strontium?
Strontium sulfide because earlier we have seen in its bulk system, its efficiency was very high. So we wanted to probe it at the nanoscale.
Okay.
So that's why we wanted to compare the bulk system with the nanoscale.
The reason is that you are doing the up conversion and we are using cerium,
Up,
europium,
yeah,
erbium,
up
all are
conversion,
the-
up conversion we have not done with strontium sulfide.
Yeah, that's what one more question I have, that strontium is also giving up conversion, or we are doing doping of erbium, ytterbium, terbium, they are giving the up conversion.
Yes, yes. It's see, as far as the luminescent scientists know, there are certain pair combinations which gives the up conversion. But many a times, even a single dopant, which is having energy levels compatible with the host system, they are in the paper we have seen in the literature. Even a single dopant sometimes give you up conversion.
No, he has done erbium, europium, terbium.
In combination, we have done.
But the point is that erbium, europium, terbium is very costly if we will go for the up conversion studies. Can we try for magnesium? That I mean,
No, it's not possible because the resonant energy transfer is you have to see the energy level diagram.
Okay.
So you have to tune the host materials in order to see these transitions in the low cost dopants.
Thank you, sir. Thank you so much. So I will, yeah. Okay, so as we begin, okay, as we begin this auspicious conclusion ceremony, the valedictory sessions, we invoke divine blessings before proceeding further to create an atmosphere of positivity, wisdom, and the harmony. We now present a graceful Ganesh Stuti dedicated to Lord Ganesha, the remover of obstacles and the symbol of knowledge, success, and new beginning. So can we have a Ganesh Stuti?
गणनायकाय गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि.
Ladies and gentlemen, We now formally begin with the valedictory ceremony of International Conference Past SG 2026. This conference has been a meaningful academic journey, bringing together eminent scientists, faculty members, industry experts, research scholars, and students on a common platform. The discussions over these 2 days have emphasized the need for interdisciplinary research, sustainable innovation, responsible technological development, and stronger collaboration between academia, research institution, and industry. This conference has not only provided a platform for sharing scientific knowledge, but has also encouraged young researchers to think beyond disciplinary boundaries and contribute towards solutions for real world challenges. Now, I would invite all the respected dignitaries to please come on the stage. Professor Anirban Basu, sir, please. Professor Seema R. Pathak, Professor Rajendra Prasad, sir, Professor Preeti, Professor Sudeshna, ma'am, please come on the stage. Sudeshna, ma'am, Preeti, ma'am, Seema, ma'am.
Okay.
बोलो मैम बोले मैम मैम आप
So thank you, our very own RP sir, and we welcome you on stage. Sir is leaving us. So this video was conceptualized between me and Asim, Dr. Asim, and I told him to take pics during we are doing it and preparing it, and he made it. I think we must clap for him. एंड डॉक्टर असीम टोल्ड मी कि मैम जिसका फोटो नहीं होगा तो मैंने कहा कि फिर पिटने के लिए तैयार हो जाना क्योंकि काम तो सबने किया है एंड आई थिंक ही इज नॉट इन ऑडिटोरियम ही लेफ्ट अच्छा यू आर सिटिंग देयर कम हियर कम safer position. So you all may be knowing him. He is very popular face, Faculty of Forensic Science, our own PhD. He did PhD from here. Manju ma'am must be knowing. And I must thank Manju ma'am and Dr. Sangeeta ma'am for gracing the occasion at this valedictory session. And now hand over to this one, Dr. Pooja.
Thank you so much, ma'am. So again, I will invite all the dignitary, please come on stage.
Ma'am and this.
Okay. One of our speakers, she was not able to connect earlier. So before proceeding to the prize distribution and valedictory ceremony, we would want to invite our speaker. I hope she is here with us. Dr. Ayushi, are you able to listen?
Yeah.
Okay. So, we shall now formally invite Dr. Ayushi Arora, Assistant Editorial Development Manager, Royal Society of Chemistry. She is associated with the Royal Society of Chemistry's India Publishing and Editorial Development Initiatives. She has been involved in activities that support authors, researchers, and wider chemical sciences community in improving research visibility and publication engagement. May I request Dr. Ayushi to kindly address the audience?
Yeah. Thank you so much for the nice introduction. And at the onset, I would like to thank the organizers for giving me an opportunity to connect with this audience. And I have a few slides to share. So I think it'll be better if I start sharing. Are my slides visible?
Yeah, it's visible.
Yeah, okay. So as it has already been introduced, I have been associated with RSC since 2023, and I would like to walk you all through some of the initiatives of Royal Society of Chemistry in connecting chemical sciences community. So before we begin, I have a small video to share, and I think it will sum up what RSC does in India and globally.
From the birth of the Industrial Revolution to the future of sustainable prosperity, the chemical sciences are at the heart of human progress.
The pace of change is incredible, driven in part by the amazing discoveries and advances in science and technology.
The Royal Society of Chemistry's purpose has always been to advance the chemical sciences. We champion the chemistry profession, setting standards and celebrating excellence.
We share chemical knowledge, publishing the best research, and bringing scientists together to connect and collaborate. And we use our voice for chemistry, speaking up for the chemical sciences in the public interest.
We are a community of 50,000 members across the globe that celebrates our differences, working together with a common purpose to realize a better world.
Our purpose is unwavering. But as the world changes, we constantly evolve how we advance the chemical sciences.
Science is an international endeavor, and we live in an increasingly globalized world.
We've expanded our presence from our historic London headquarters and Cambridge hub to a network of offices across the world, from Beijing to Berlin, Shanghai to Bangalore.
Technological advances continually raise scientists' expectations of what is possible.
We use advanced technologies and data science to help researchers unearth and share knowledge faster and more effectively than ever before. And we bring together visionaries to explore how digital technologies will allow us to discover unexplored frontiers and to make sure the next generation of chemical scientists are equipped for the future.
As a professional body for chemistry and a champion of chemistry professionals everywhere, we've broadened our horizons and recognize an increasing diversity of skilled and passionate people.
As society recognizes and begins to tackle many social injustices and discrimination, we are proud to have bolstered our own research and action to make chemistry a more inclusive community, accessible to everyone.
These ever-developing factors of globalization, technology, and inclusivity have a huge impact on the way scientists need to share and access research. In response, we'll make research more open and accessible on a global scale. As society changes, chemistry must change also.
Historically, the Royal Society of Chemistry has successfully adapted to support our community's shifting needs.
And will continue to look to the future and ensure everyone can benefit from all that the chemical sciences will bring to society.
Let's advance chemistry together.
So as the video summarizes, we have several offices. One of them in India, we have two. Actually, we are based in Bangalore and also in Gurgaon. We have a network of nearly 60,000 RSC members worldwide. And India team is actually a very small team comprising of 14 members altogether, including sales, publishing, and engagement or membership. So we are a non-for-profit international publisher and are indexed in several services using ChemSpider, in Merck Index. This is just a a slide just to show that how diverse the Royal Society of Chemistry journals are, right from analytical chemistry to environmental sciences to energy to drug discovery and delivery. We had several new launches over the years to cover all spans of chemistry, and which include both hybrid as well as gold open access journals. So for example, this is just a representative slide for all our gold open access journals covering different UN sustainable development goals, as well as connecting chemistry with the interdisciplinary sciences. So not just the journals, we do publish several periodical reports and books. So this is an example of what specialist periodical reports were published from India. Also, we have a very diverse collection of e-textbooks which is available in every single category of research, for example on nanotechnology to organic chemistry, as well as there are several Indian authors who have contributed either as an editor or an author of the book. Some of them are listed below. And in that particular respect, if you think you have an idea in mind and there's a gap in research that one should address to and would like to contribute as an author or an editor, so please feel free to reach out either to the books team or to us, or you can also scan this QR codes to submit your query for any book for contributing as a book author or an editor. So there are several other resources which are also available online for teaching, ranging from primary to higher education, and all these are available on our website. So as we all know, India is growing in terms of research. So the trend continues in 2025 as well, and India is the second largest contributor to submitting to RSC as well as getting their content published. First being the China, and that this is just growing year by year. We have a network of board members across several journals across India, right from Kaushik Chatterjee, who is on board with us in JMCB, to Rekha Singhal, who is an associate editor for one of our newer journals, which is Sustainable Food Technology. And not just these, we recently expanded our editorial board with Indian editions, and one of them is Professor Sandeep Murarka, who joined us on RSC Advances as an associate editor from IIT Jodhpur. Now, not just the editorial board members, we do celebrate the milestone celebrations of institutes as well as authors. So in the honor of Professor Amitabha Patra's 60th birthday, we had a collection in nanoscale, a cross-journal collection across nanoscale and PCCP, which was guest edited by 4 of our editors, and those are listed here. Not just this, we do celebrate the milestone of events as well. So the 75th anniversary of IIT Kharagpur, there is a special issue marking the scientific excellence. So if there's any idea or any themed collection plan that you have in mind, so you can always reach out to RSC for collaboration on that. And then we also have several of our events where we try to connect the audience, the researchers, both at early career level and established researchers, with our editorial board members and our experts in the field. So one such example is which happened in February recently in 2026 at IIT Bombay, where we had a meet the editor session in which Professor Burt and Professor Karen, who are the EICs on 2 different RSC journals, joined us along with several other experts and board members, and we had a very interactive session where students and faculties participated. So we do them around the year in different locations in India, and the other one is like when we had a series of Meet the Editors in November 2025 at IIT Jodhpur, BITS Pilani, and Naipur Mohali. So these are some of the snapshots from other similar kind of engagements that we had at NIT Patna and the PCCP Symposium. So one of our flagship event is Chemical Science Leader in the Field Symposium. Happens every year. It's an annual symposium. So in 2024, it happened at ICER TVM. And in 2025, we brought the symposium to ICER Pune. This year, symposium is in planning, and the best part of the symposium is that you get to meet the editorial board members, the associate editors who are judging your manuscripts on the chemical science journal, as well as experts from from the global community who are working on different aspects of research. So, and then we have the best poster prizes, but the good thing is that some of the best poster prizes get to present their research orally at the end of the day, at the end of the symposium, like on the last day of the symposium. So that's one of the exciting features of the ChemSci Leader in the Field Symposium. So not just these, well, to publish in journals, you obviously need publishing resources available. So we have several of the resources available online under the authors and the reviewers tab, which include how to publish your research with maximum impact. So this is available freely. You can download. And also the next thing that is available is, of course, once you have published, you all would also like to review for RSC. So how to be a reviewer for RSC, including step 1, where you you check your eligibility. Secondly, you kind of you look at the reviewer procedure and policy for the guidelines, and then you start your application, submit, and then you hear from us that if it has been approved or not. Now, not just journals, books, categories, events, and everything. RSC does offer several levels of membership depending upon the research stage that you are, right from affiliate membership, which is available to students, and to the Fellow of Royal Society of Chemistry, which is the FRSC. Now, one benefit to become, as for students especially, why should you join as a member for Royal Society of Chemistry? Because this gets to actually make you eligible to apply for several grants which are available to the members of RSC, and one of them is a research fund grant where you get up to £5,000 to fund your research for Royal Society of Chemistry. And then the second is researcher collaborator grants. So this is available throughout the year. You can submit your application, and the money you get, you can use it for for going to a collaborators institute for research.
Sorry, ma'am, please finish it in 2 minutes.
Yeah, I think I'm done. Sorry.
Thank you so much. Thank you so much. Now I would like to invite all the dignitaries on the stage. She's about to finish, she said.
Yeah, I think I stopped sharing.
Thank you. Thank you so much. Please, ma'am, please, our HODs, Professor Preeti Thakur and Professor Sudesh Naghosh.
बच्चे पीछे जाना है सब चाय को सर्टिफिकेट नाम तुम्हें और शुरू कर दो सर आएंगे नाम पे अनाउंस कर दो बच्चे आ गए आओ आओ प्रतिभा आओ प्रतिभा
Okay. Professor Basu is here. वही था ना आपको बोलिए
Before going ahead with the prize distribution ceremony, we want to felicitate our guest of honor, Professor Basu sir. And it is our honor to introduce our guest of honor, Professor Basu. He is a distinguished scientist, senior professor and leading neuroscientist in the National Brain Research Center, Manesar, Haryana. He earned his PhD in immunology from CSIR, Indian Institute of Chemical Biology, Kolkata, followed by postdoctoral training in neuroimmunology at Pennsylvania State University of Medicine, USA. He's a recipient of many prestigious honors, including the National Bioscience Award, Vasvik Industrial Research Award, J.C. Bose Fellowship, and the Haryana Vigyan Ratna Award 2024. Now, may I request the dignitaries, Professor Seema R. Pathak Madam, Professor Preeti Madam, Professor Sudeshna Madam, to felicitate our guest of honor. Now we request our guest of honor, Professor Anirban Basu, to please deliver your visionary address.
Hello. So very good afternoon, all of you. And I want to thank Professor Pathak for this kind invitation and all the dignitaries in the dais and all my fellow colleagues from Amity University and other universities. And of course, dear students. So when I first got this invitation to come here for this meeting, I thought, what actually sustainable development mean? So any of you know what is the difference between sustainable development and development? Any of you, any idea? Well, why this sustainable development is so important for a country which has 1.4 billion population? Because as you see our Prime Minister's vision to make India a developed country in 2047, if you really want to make this country developed, that means you have to bring your big amount population above the poverty line. So still our good amount of population is below poverty line. We have to increase their living standard and make them better off in their life and of all aspects of life. And in that aspect, sustainable development is very important. Why? If you see the last talk by Professor Vij, he mentioned about this LED. LED consume very less amount of electricity compared to the regular bulb. And if you wanted to light all the household in this country, then you don't have choice than you take the options of LED. Because if you do regular bulb, they need lots of electricity. And as of today, we know what's the importance of the energy. There is a war happening somewhere else, and entire country, we are facing the heat of LPG, we are facing the heat of petrol, we are facing the heat of diesel. The reason is same, energy. So for this 1.4 billion population, you need huge amount of energy to sustain and to develop. 20 years back, we didn't have any concept of inverter AC. Today we have inverter AC. Why we have an inverter AC? That can consume less electricity. So you understand the fundamental point of sustainable energy. Sustainable energy, because you need energy which actually you can provide development for the entire population. That's why your development should be sustainable. And now we have electric car because we know that fossil fuel is limited. One day will come that we won't have any fossil fuel. Then how will you run the car? Then we have a hydrogen-based car. Now it is reality. Several Indian Institute of Technologies, they have this technology now though it's preliminary stage now, but we can think about a hydrogen-based car, electricity car, of course we have. So another important thing is the agriculture. We now, our Indian Council for Agricultural Research, they are much more advanced research they are now doing, how we can have rice or wheat which need less water. Water is another source of energy which also not unlimited. We know the place we are here, entered Gurgaon, Haryana, this part of Haryana, our water level is depleted. And you don't have water if you go to the surface for many feet. So we have to develop cultivation which need less energy. And I'm not sure how many of you know in Rajasthan, some part of Rajasthan, already based upon some technology from Israel, we are having farming or agriculture which is depending on the less water, because Israel is a country which doesn't have hardly any water. And they are now cultivating the olive in Rajasthan desert with help of this technology from Israel. And India also start making olive oil based upon this olive cultivation in Rajasthan desert, and very less water they need. And it's totally a technology which Israel developed. And also we now understand how to make fresh water out of seawater. This is also technology which Israel is sharing with India, because they don't have fresh water sources. From the sea water, they have to make drinking water. And if you think about our population, we cannot probably afford to give the drinking water to this population for many years to come. So we need to have a technology that way we can actually recycle seawater for drinking water. This is a sustainable development. Because particularly when your population of a country is say 10 million, 12 million, 15 million, like lots of European countries, you don't have to bother that much. But when you have a country which is a 1.4 billion population, more than development, sustainable development is so key. Hope I am able to explain why is sustainable development so important. And I'm sure there are many talk you have listened in last 2 days, which are definitely about the green energy, how chemistry can be used, how physics can be used for sustainable development. With that, I want to thank you, all of you, and very good wishes for your next endeavor. Thank you so much.
Okay, so now we are moving towards the last session, award ceremony. But before we moving towards the award ceremony, I would like to invite Professor Ankush Vij. Sir, please come for the felicitation. I We'll request Professor Basu and our HOI, Professor Seema Pathak, our HODs, please come. Next, I would like to invite Professor Gyaneshwar Rao. Please come on stage for helping so much. Yeah, please. Yes, ma'am.
So I must say that I'm blessed that I have wonderful faculties. Right now, I will say in vote of thanks, but this efforts RSC was by Dr. Gyan. He just convened this award ceremony with RSC, and the one lady came yesterday, and today the session was there. So I'm thankful for that. And you must not be knowing, students, he is our adjunct faculty now. He has not left Amity completely. So you can call him with all rights anytime for his help. Okay. So thank you so much, Dr. Gyan. Come here. So we are very good team. Thank you so much.
As ma'am said, our awards are sponsored by the ACS and RSC. Now I will announce the name. For the ACS awards, Maths Oral, Steppy, AUH. Dr. Gyan, please come. Ankush sir, you can also come. Please, you can also come. Okay, next we have Maths Online, Tanmay Sen, AUH. Come with supervisor. Where is? Online. Okay, he's online. So, okay, next Physics Oral, Nandisha Alawat, MDU AUH, I think. Okay. Okay, now in online we have Bhavna Vidhi. Online. Okay. Chandigarh University. Forensic, we have Khushi, AUH. Forensic Khushi. खुशी बहुत खुशी हो रही है पर खुशी कहाँ है फ्रॉम फॉरेंसिक AUH. अच्छा तो फिर यही है भाई. हाँ खुशी रिपोर्ट अब RSC है
So 5 award has been sponsored by RSC. So first 2 winner will get 5,000 each Amazon vouchers, and last 3 winner get 3,000 each Amazon voucher. The first award goes to Akansha Shoni from MNIT Jaipur.
Akansha Shoni, MNIT Jaipur.
So
online.
I will request Dr. Kamal to
Okay, Dr. Kamal, you can come, please.
collect the award.
On behalf of Akansha Soni, you can collect the award. Yeah, MNIT Jaipur.
The second award goes to Jyoti, Amity School of Applied Sciences.
Dipti ma'am, please come. Supervisor, which supervisor?
The third award goes to Ripun Yadav.
Ripun Yadav. है तो ये कौन है?
Ripun.
Ripun Yadav, AUH. Oh, Supreet ma'am, please come.
The fourth award goes to Sangam and Nikita from SGT University. Sangam and Nikita.
Okay, Sangan and Nikita. एक SGT.
SGT. Yes, yes. हो गया.
Okay.
दोनों है.
Sangam and Nikita.
And fifth award goes to Rahul Yadav.
Rahul. And we have two supervisors, Manish sir and Dr. CM. Please come. No, please, both. Mr. Rahul, be fast. Please come.
इनके बोले सर्टिफिकेट दे दो ना यहाँ पे वहाँ पीछे दे दो संगम इस साइड
All RSC award winners are requested to share their email ID.
Even ACS award winners, all ACS and RSC award winners are requested to share your email ID.
Thank you.
ACS awardee will get 1 year membership. You can open all your journals. Yes, sir. Thank you so much, sir. Now, the last part, I will request our HY Professor Seema R. Pathak to give vote of thanks.
So this is a time of, maybe we are doing it from last 2 days, but no, we are doing it from last 4 months. And we thought that we are late in planning, but no, we had completed it successfully. So I have very, this is very emotional moment for me because it was, great achievement. It is a great achievement for me as a team. And now I will propose vote of thanks. So our profound thanks to our leadership of Amity. We are deeply indebted, Dr. Ashok K. Chauhan, sir, Honorable Founder President. You clap for everyone's name. Amity Education Group, for his blessing and vision that continue to inspire us to pursue excellence in science and education. We express our sincere gratitude to Dr. Asim Chauhan, sir, our Honorable Chancellor, Amity University, Haryana, for his words of wisdom and constant encouragement. Maybe he was not physically present, but actually he was with us and motivated us to host this event on international stature. Our heartfelt thanks to our Vice Chancellor, Professor P.B. Sharma, for his inspiring address and academic leadership that he spread for FAST SD 2026. We are grateful to Professor Dr. Vikas Madhukar, our Pro-Vice Chancellor, Dean FMN Director ABS, for his warm welcome address and for extending wholehearted institutional support. To our distinguished chief guest and guest of honor, we are honored to have presence of yesterday we had this Dr. Professor Diwan Singh Rawat, Honorable Vice Chancellor, Kumail University. And then we have got guest of honor, Professor Govind Gupta, Chief Scientist, CSR NPL. Today we have Dr. Pratishtha Pandey, and we have on dais Dr. Anirban Basu from NBRI. So we are grateful that they have spared their time. And I also thank to our speakers from this Pramod from Thailand, and then this Nades, this Moloki Tinia from Russia, and both have done wonderful job. Then Professor Ankush Vej for having this plenary session in post this valedictory session. Then we have a special thanks to Krishna R. Chaturvedi, Program Manager, maybe she and ACS, and Dr. Ayushi Arora, Assistant Editorial Development Manager from RSC, for their insightful session. Now I thanks to all our speakers. Of course, you had had a very elaborated session. We had 4 tracks running totally: physical sciences, chemical sciences, forensic sciences, and mathematical sciences. So I will not name off all speakers. There were total 41 speakers, and we were actually very impressed by their the way they have delivered lecture, and I hope that you all have learned a lot. Now, this main vote of thanks is for our, SAS faculties. So I will not read this text. That vote of thanks is going to be from my heart. So I will first of all, please, a huge round of applause for our co-chairs, Dr. Preeti Thakur and Dr. Sudeshna Ghosh. Without them, I was nothing. They have worked so meticulously and given directions to me also at many points. Now I will clap for my 4 conveners, Dr. Anirban Das, Dr. Deepti Vaya, Dr. Sarika Chaturvedi, Sarika Jain, sorry, and Dr. Chandrasekhar sir. I request to raise all of you please raise on your places. So actually they deserve applaud, lots of applaud, because so we have, thank you so much for being wonderful conveners. Actually, this job, what I'm doing was job of convener, but because they were 4, so I was left with no choice, and I proposed vote of thanks. So they are actually very good people, very wonderful researchers, very good administrators. So I bless from my heart that please be very good as you are right now, and may God give you lots of success in your life. Now I thank my co-conveners, Dr. Pooja Rawat. So who else? I will clap for her for 3-4 more times. And I say that actual convener was Pooja Rawat. Because she chased me too many times. Madam, आप तो conference कर नहीं रहे हो, आप तो conference कर नहीं रहे हो, और वो नाराज होकर मेरे केबिन से चली जाती थी. अब आपने फिर 6 महीने टाल दिया, आपने फिर 6 महीने टाल दिया. So in that way, she did wonderful job at many aspects, many hats on her head. So then after that, co-conveners, नाम भूल रही हूँ, Supreet ma'am, please get up. And then forensic Dr. Bhunesh, Amit. अरे मैं नाम भूल रही हूँ यार, इतना दिमाग खराब हो गया मेरा. Co-convener कौन था? नहीं, 4 थी. मैं देखूंगी.
Dr. Pooja, Dr. Bhuvnesh, Dr. Supreet और Dr. Amit.
Where is Dr. Amit? कहाँ है? कहाँ है Dr. Amit? हाँ, मुझे दे दो. So Dr. Amit का काम सबसे टफ था, because running towards all 4 sessions and taking attendance. Of course, many other support, but this I have seen working with his, Professor Dr. Amit. Thank you so much. Thank you so much. Please, थोड़ी सीटियां वीटियां बजाओ. तुम लोग को नहीं आता क्या कुछ करना? Yes. Very good. And then this our very important forte. Now it will come for this treasurer, Dr. Manish Shandilya, Dr. Leena, Dr. Dimple, and Dr. Kamal. अब आपकी बारी है, sir. Then we have this organizing secretaries, most loved one, Dr. Gyanadeshwar Kumar Rao, Dr. Tapeshwar, Dr. Niru Dabas. Where is she? Dr. Niru. Where is she? Dr. Niru कहाँ है भाई? नहीं है, चाय पी रहे हैं. Dr. Niru Dabas, Dr. Kamal Nayan Sharma, Dr. Aina Bhaduri. Very good track manager. सारे abstract. Yes, very good. Dr. Chandramohan Srivastav, very good poster manager. Dr. Vijay Kumar Yadav, very silent worker. Where is Dr. Vijay? Thank you so much. Dr. Priyanka Verma. Dr. Priyanka Verma, Dr. Vikas Laharia. So, अब मुझे गाना अच्छा याद नहीं आ रहा, बट मैं शायद अभी थोड़ी देर में मुझे याद आ जाए. कितना अच्छा बॉन्डिंग था सबका, कितने अच्छे से काम हुआ है. So then after that, this organizing committee, Professor Joydeep Datta, most respected Professor Joydeep Datta. Where is he? हेलो सर, उतने दूर क्यों बैठे हो? आ जाओ. Lots of contribution from him. Thank you so much from deep of my heart. Right? Then Professor Sunita Kumawat ma'am, thank you for having this Anirban Basu sir here. Thank you so much. And Dr. Abhilash, he is not here because of some personal reasons. Dr. Rita Bharadwaj. Where is Dr. Rita Bharadwaj? Thank you so much. Dr. Gagandeep Kaur. So, more claps for Dr. Lucky Krishnaiya. Where is she? Where is he, Dr. Lucky? Whatever slides you are seeing on his stage, it is made by her. All 40 slides. Great job done. डॉक्टर आयुषी ऑफ कोर्स जो सबसे साइलेंट होता है वो रिपोर्ट राइटर होता है और उसके बिना कुछ नहीं होता है. सो डॉक्टर आयुषी, डॉक्टर संजीव चौहान फॉर मेकिंग वेरी इंपॉर्टेंट थिंग्स, फूड कूपन्स, डॉक्टर मिस्टर अजय, डॉक्टर संजीव चौहान एंड मनोज. सो वंडरफुल जॉब डन. जय श्री राम. डॉक्टर ममता पाल फॉर रजिस्ट्रेशन. वेरी गुड. जोश आई है. डॉक्टर नीरज, डॉक्टर असीम फॉर मेकिंग सच अ वंडरफुल वीडियो. डॉक्टर सार्थक फॉर कंपाइलिंग दिस. डॉक्टर सार्थक, वेयर आर यू? गुड जॉब डन. एंड देन अभी तो कुछ और हैं जिनको फैकल्टी को पहले बोल दूं जिनका यहाँ नाम नहीं था. इसका नाम नहीं था. डॉक्टर सार्थक सबका हो गया? नहीं, अभी नहीं हुआ. डॉक्टर, yes. Now I will acknowledge my faculties, those who have sponsored funds for this event. First of all, Dr. Anirban Das, Dr. Gyandeshwar Kumar Rao, Lab India and Agilent. And then Dr. Anirban Das, again another fund. Then Dr. Pooja Rawat awards and mementos. These all were arranged by different vendors by them. Dr. Leena, where is she? Dr. Lucky and Dr. Supreet. Please clap for them. All of them arranged so many things for this conference. And then after that, I mean, who is the most important, for whom we clap a lot? That is my dear students. Because without you, that nothing was possible. Lots of patience you guys have, because having this type of session is sometimes becomes quite boring. So thank you so much for being wonderful participants. And then मैम ने बहुत अच्छी बात कही. वैसे मैं अजय और मनोज को थैंक यू बोल चुकी हूँ. लैब स्टाफ मिस्टर प्रमोद, मिस्टर श्याम सुंदर and Mr. Kuldeep, Miss Radha and Mr. Dilip. Yes, they have worked a lot. Miss Sheila and सबसे ज़्यादा important तो शिव कुमार, अजय, शिव कुमार, मनोज, इनके बिना कुछ possible नहीं है. So keep working in that way and thank you so much. सबसे ज़्यादा मैं किसको अभी thanks कहना चाहती हूँ? श्रीनिवास. Please come on stage. श्रीनिवास जी हैं यहाँ पे. Vivek, Srinivas ji and Vivek Gautam from Amity, Noida. Srinivas ji and hospitality team. Hospitality team, Ravi Chahal sir, Rear Admiral sir and SK Singh sir के office से. Of course from this accounts office also. Where is Srinivas? Srinivas, आओ ना एक मिनट. आओ एक मिनट. ग्रुप फोटो भी होनी है, भाग मत जाना भाई. श्रीनिवास सर आ जाइए. भाग गया श्रीनिवास. So I think all of us is बैठ जाओ. वो तो वोट ऑफ थैंक्स में ऐसे ही होता है.
We have a group photograph. Please stay.
ग्रुप फोटोग्राफ जरूर करा लो भाई. वेरी श्रीनिवास, वो मुझे दिखे देना. कुछ छूटा तो नहीं आपको पहले. एंड मुझे कौन थैंक बोलेगा? आपका वो है ना कौन? अच्छा, so all faculties are requested to come here. I will request Dr. Ankush to give us that one. And sir is there also. Ankush, आ जाओ एक बार stage.
Please, all the faculties, all SAS faculties, please come on stage.
SAS faculties, सभी लोग आ जाओ फटाफट.
SAS faculty, please come on
Dr.
stage.
Mamta, everyone,
I
please come.
request Dr. Ankush and Professor Basu.
All SAS faculties are requested on this stage. Dr. Supreet for memento, बहुत अच्छा काम किया.
Vikas
जल्दी आओ
ma'am,
sir.
please come fast.
Anirban sir इतना slow चल रहा है, जल्दी आओ.
Vikas ma'am, please come fast.
Sunita ma'am.
Dipti ma'am. Sunita ma'am, please. आओ. फोटो फोटो फोटो. उसको बोले फोटो प्लीज.
Student volunteers.
आओ. असीम. आओ.
ऊपर आ जा.
आओ जल्दी जल्दी कम फास्ट.
ज्यादा गेम कौन बन रहा था? खुद ही वीडियो बनाएगा, तेरे वीडियो बना. प्रियंका मैम.
हाँ, चाय के लिए ले जाओ, बच्चों को मैं बोल देती हूँ. थैंक्स. कहाँ गया यार? पूजा खोल ना पेज. बच्चों का नाम. Yeah. अच्छा, अब प्रीति मैम is saying, say 3 cheers for Amity School of Applied Sciences. Hip hip, hip hip, hip hip. Jai Vigyan, Jai Hind. Correct? Now I will say thanks to my students. Jai Esas, Miss Himanshi Verma, Mr. Rahul, Miss Anmol, Mr. Piyush, Miss Sarika, Miss Pooja Shravan, Miss Pooja again, Miss Mannat Kaur, Mr. Mukul, Mr. Rohit, Miss Geetika, Miss Tanu, Mr. Aryan, Miss Khushi, Miss Zia, and Miss Preeti. Thank you so much, Mr. Arvind, IT team from AU Noida. मैं काम सोचती हूँ ना कि बच्चों को मैं पर्सनली मेल कर दूंगी एक बढ़िया सा. मुझे जो जो बच्चे किए हैं ना, चलो ये ले लो, अब सामान ले लो, हो गया. बस फ्री हो गई.